Thank you so much, Professor Kasahara, for hosting my talk here and inviting me to the University of Tokyo. Thank you so much, Professor Suramichi, for organizing my talks in different places at the University of Tokyo. So I'll be starting now talking about the talk of today. But before that, let me introduce our research group a little bit. So this is our group led by Professor Onur Mutlu, and we have a pretty large number of members who keep coming and going. So we have a large number of interns who join us at different levels, as well as visiting professors. So our motto is Think Big, Aim High. And we are currently located in Switzerland, in the beautiful city of Zurich. This is the city center, and this is ETH. And we are part of the computer science department. So Professor Onur Mutlu was a professor at CMU for about 6 to 7 years, and we moved recently to ETH since September 2015. And our research spans many directions, many research areas, including computer architecture, optimizing the VRAM, the memory, and the CPU itself, and the hardware-software cooperation, hardware security, and bioinformatics as well. So we have openings. We are hiring students at all levels, postdoc, PhD, masters, interns. So feel free to contact me directly or Professor Onur Mutlu or apply to our website. Okay, so this is a personal story, and we used to receive these packages in my home country, where I received my school education and bachelor degree through JICA agency. Those packages contained personal pictures, some motivation letters from people, Japanese people, sent to the best 2 or 3 students in each class in Gaza City or in Palestine. And this really touches our heart. So thank you so much for sending those packages and supporting the education in Palestine. Okay, I'll be starting my talk now. So do you have any question? All right. So this talk is not about how to use wet lab methods or algorithm or technique. It's more or less talking about the bioinformatics side or the algorithm development rather than the wet lab procedure. So I'll be talking about why we picked genome analysis as a problem to be accelerated, and what is genome analysis basically, and then we talk a little bit about read mapping problem, and then present the algorithms and the hardware accelerator or architecture that we developed to accelerate this problem. And towards the end, I'll show you the future of this area, whether the read mapping will still there for 10 or 20 years later on in the future. Okay, so let's start why we bother about genome analysis. So I can give you hundreds or thousands of examples show the importance of genome analysis. These are just few of them. For example, personalized medicine, GWAS studies, city-scale microbiome profiling, tracing birth parents, for example, want to know who's your father, who's your mother, and so on. Or disease risk profiling. You don't have any diseases, but you want to know in the future, will you have certain diseases or not, for example. And many more. So probably you know this term, which I can't pronounce it. So this means difficult illness, which is a term coined by the Japanese Ministry of Labor, Health and Welfare in 1972. And this is actually the correct term to refer to rare diseases. The word calling it rare disease, but actually it's not rare. It could be difficult. It's the more accurate term. So the rare disease is defined as 5 or 1 in every 10,000 population having that disease. So in the world, it's expected that 1 in 17 people worldwide to be affected by a rare disease at a certain stage of their life. That is translated into numbers to 350 million people. And 75 of them are children. 30% of these children never make it to their 5th birthday. 80% of these people having a rare disease because of genetic disorder. So that shows you the importance of having personalized medicine that can address these genetic disorders. And in Japan, Professor Matsuda, for example, from Kyoto University, says that we don't know exactly how many people in Japan having a rare disease. But you can think about it as out of 20 of your friends, one of them will have a rare disease at a certain stage of his life. For that, Japan started an initiative to address rare diseases, or at least have some statistics, some knowledge about it. And they spent for that around 600 to 700 million of Japanese yen a year to read the genome of more than 2,000 patients every year. In UK, for example, starting from this year, whoever seriously ill children will be admitted to a hospital in UK will be offered directly a whole genome sequencing as a part of their inpatient. So that, by using whole genome sequencing, we are expecting to reduce the inpatient cost by 100,000 to 300,000 US dollar. So think about the money you can save in Japan, for example, by doing whole genome sequencing to all children, or critically ill children, as well as the adults, for example. So if you have a database of all the individuals in Japan where you already know their genome or the causes of certain diseases, then you can manage the things pretty much well than what we do these days. So the second example of the importance of reading our genome is GWAS study. In GWAS studies, we have two groups of people. One of them, we call them healthy, and the other one unhealthy, or having certain disease. And by healthiness here, we're not necessarily targeting a disease. It could be an eye color, hair color, and so on. So what we do with these two people, as we are guaranteed that this group having the same disease and those they do not have it. So when we compare their genome, here we have the x-axis, for example, which shows the chromosomes of the human genome, and at each location we draw it on the logarithmic scale to signify the different, to have a significant difference between these SNPs, between these individuals. So whenever you have this peak, this kind of peak, meaning that at this location, with chromosome 11, for example, you have a certain SNP that is correlated with this disease that you are targeting. So you can guess why we are calling this plot as Manhattan plot. So here's an example. For example, you can notice the blood pressure, and this is each row representing an individual or a portion of individual genome. So each row is separate human, and you can see we have SNP1 and SNP2. So you are smart people, of course, and that's why you can guess that SNP1 is correlated with blood pressure. Why is that? Because whenever you have C at this position, meaning that you will have most likely a high blood pressure. But for SNP2 is not correlated to that because you can see just some random values. Okay, so do you think always single SNP what causes disease? This is not the case, unfortunately. So for example, these diseases are caused by large number of variation in the genome. For example, if you deleted 500,000 of characters from your genome or from the SNP, you will observe these two diseases. However, if this gets duplicated, the same amount, duplicated in your genome, then probably you'll have one of these two diseases, which are the opposite side or the opposite coin of these diseases. So these are well-known paper published. This is the reference of each of these diseases, showing that whether you will get deletion or duplication of this portion of DNA. So you can see it's really challenging. We are not always targeting single SNP. It could be really huge to find these deletions or duplicated. Okay, this is the third example. I'm talking about here city-scale microbiome profiling. So in this example, think about very crowded places where you would like to know what kind of bacteria or viruses you have in this crowd. Think about Tokyo subway station, for example, or New York subway station. It is really harmful to find some harmful bacteria in these places because it can affect a large number of people. So for that, this paper in 2015, they target New York subway stations, all of it, and they collected around 1,500 samples where they do bioinformatics analysis and try to analyze what do we have in these samples. So they swap everything, including the train seats, the doors, the machine for the card, and so on. And what they found, around 50% known bacteria, and the other 50% unknown organisms. They don't know what they are. And the interesting thing in this study, we found the black. So in this disease caused by Yersinia pestis bacteria, and for those who doesn't know this disease, this is in the 14th century killed more than 1/3 of the population of Europe. So you can think about how harmful this disease to find in New York subway station. And this raised a lot of attention in the media in the US. And they were thinking about closing all those stations until they investigate the situation there. However, they find it as a false alarm. The Yersinia pestis bacteria, it wasn't there. And some people call this a failure of bioinformatics. However, this is not true. I don't agree with that. Bioinformatics is still doing really great. This is a problem with the tools they use in this study. And this shows you the critical need, not only for fast, but you need really accurate genome analysis tool. So you can't compromise the accuracy of the tool if you want to accelerate it. Acceleration alone is not enough. You need to maintain the accuracy of the tool as well. Okay, so I'm going next to talk a little bit about what is genome analysis actually. We already showed the importance of accelerating this problem. Okay, for genome analysis, you have a genetic sample. Let it be a blood, for example. I want to convert it into a text format. So this is normally a chemical format. You can't do anything about it using computer algorithms. So what you would like to get is the full text of the human genome, for example. And then after you're getting the full text, you try to compare it with someone else's genome to find some differences in that genome. However, we don't have such machine that get the genetic sample and give you the full context of your genome, unfortunately. So, I'm pretty sure that most of you took maybe biology class in school, so this is a little bit like very beginning level of biology. This is our human cell where we have the DNA stored here, and we have many chromosomes. And in our cells, we all the body cells having the same content of DNA. So it doesn't matter which cell you're trying to analyze. It could be blood or hair or skin tissues and so on. All of it having exactly the same content. But what it makes it a nose cell or tissue cell or hair cell is how you activate, think about it as switches. If you activate some of those switches in your DNA, then it will be converted to something else. So we started always by the DNA, which is, we call it a genotype, that you can think about it as a recipe book. You have a cookbook and you want to cook something, for example. And then the DNA gets transcribed into RNA, and you can think about the RNA as the recipe to cook something. So this is including all recipes, and this is a single recipe, for example, you would like to study it. So the RNA gets translated into protein, and then protein give you the phenotype or the food you'd like to cook. So from the protein you can observe the eye color, the hair color, skin color, and so on, or anything that is visible to you. So the cause of this phenotype is going back to the DNA. So that's why we'd like to study the DNA. And the DNA has some chemical molecules which we refer to them as A, C, G, T, or adenine, thymine, guanine, and cytosine. So A always binds with the T using 2 hydrogen bonds, and the G and C with 3 hydrogen bonds. And this is what causes the double helix structure of the DNA having the A and C and G, T. So if you stretch the DNA out of the cell, it will be 2 to 3 meters long. And the DNA is normally supercoiled such that it can fit in a very tiny place in your cell. Okay, so how long is the DNA? When we are talking about viruses, it could be a few thousand. When we're talking about bacteria, it's a few million. And this virus, by the way, can affect this bacteria, causing some disease. And for human genome, it's around 3.2 billion. Do you think we are the best on the earth? Of course not. The red onion having like 5x longer than our DNA. And the Japanese flower having around 15x longer than our DNA. Okay, So for genetic similarity, when we compare human to human, we observe 99.9% similarity, and we are always looking for only 0.1% differences. So your tool should be very sensitive to capture that difference. And if you mess up with this similarity and differences, you may end up comparing a human to a banana with 50% of similarity. So this is our chromosomes under microscope, and this is chromosome number 12 for Henrietta Lacks. Does anybody know who's Henrietta Lacks? Okay, so this is the girl. So this girl, her cells were distributed all over the world, and they were the seed for all the genetic studies. She was diagnosed by cancer in the very old days, and her doctor shared her cells among all research labs without her awareness. And they made a movie about it, so it's better to watch it and read about Henrietta Lacks. That's why they call it the immortal life of Henrietta Lacks, because she died, unfortunately, because of the cancer, but her cells are still used in today's lab. Okay, so from the picture under the microscope, you can see that chromosome problem is, or reading the chromosome, the content of the chromosomes is really similar to the yarn balls. Think about having those yarn balls, and they are really they are tangled, so they are very complicated, and someone asks you to unstretch it out or untangle it. So what's the optimal way to do it? You don't have any option unless you go and cut all the tangles there until you get a very smooth string of the yarn balls, right? Because it's really complicated. If you spend time trying to remove the ties, it will take you some really long time. And this is what we do for reading our DNA, because it's really complicated, really super coiled, and it's very small level. And that's why it took us 13 years to read the first draft of the human genome, and it cost us $3 billion. So think about each character of the human genome cost us $1. And the first draft was in 2003 with the collaborative work between Japan, UK, US, and some other countries. However, in these days, we are doing really good in reading our genome. And we could read 1,000x more bases than before in only 44 hours and less than $1,000. However, what we got is short pieces of our DNA. It's not the complete content of our DNA. It could be short, it could be long, but at the end we're getting pieces of the DNA. It's not the full DNA. And we refer to the machine giving us this information as sequencing machine. Sequencing means here reading the content of the DNA, for example. And these are the sequencing machine. We are calling them sometimes chopper because they are chopping your DNA. They are not giving you the full content. And you can think about hand-sized sequencing machine, which is a USB stick. You can plug it into your laptop or attach it to your iPhone. It could be a room size or fridge size. All of these are sequencing machines give you pieces of your DNA with different properties such as read length, the sequencing errors, and so on. Okay, how we do sequence our DNA, for example. So think about it, we have a surface, a glass surface, where we attach a DNA molecule to it, and then we try with the 4 bases. So we have A or C or G, T, right? 4 bases. So we pick the first one, for example, T, and then we add it to the surface. And then if we have A at the surface, then it will chemically react with the T, right? Because this is unstable, and using 2 hydrogen bonds, it will bound to T. If we observe a chemical reaction here, then there's an optical sensor. We'll capture the reaction using some method, and then it will tell you it was A, because when you add a T, it was a reaction. So we keep adding all those molecules and observing the chemical reaction, but we do this for A, C, G, T. And we do this after washing the surface. So we repeat this 4 times for each base attached to the surface. But we do this at scale, so that's why we call it high throughput sequencing, because we do this for many DNA molecules. And then the sequencing machine will provide all of these. However, this method for short reads, for long reads, is a little bit different than this method. And the problem with this method that the read produced by the machine has no information about the order and the location of each piece. So the piece is just string of text, A, C, G, T characters. It doesn't tell you where it's located in your DNA, or their number, or their order, which one comes after which. And for that, we need a genome analysis where we pick these pieces and try to help stick it together and build our genome, and then we do some analysis, and then we send it to the doctor for scientific discovery or whatever. So think about this as the bioinformatician, and having all these pieces coming from the sequencing machine, trying to stick it together to build the original piece or the original picture. So it really sounds like a puzzle problem where you'd like to solve it. And if you don't have a reference, or if you don't have this picture in mind, you can't just stick these pieces together. And this is what you do as a de novo genome assembly, where you're trying to just heuristically align these pieces together to build a graph, and then resolving some loops in this graph until you get a very smooth piece of DNA or genome. And it could be really complicated. For example, this is for one of the viruses, which is, though it's really small, but you could get really complicated one. So for today, I will be focusing on reference-based genome analysis, not de novo assembly. And as I mentioned, this sequencing machine could give you very small pieces, so the problem become harder for you, or large pieces. However, which one is better for you, smaller or larger? It sounds this is better, right? Because it's larger pieces, so you can solve the puzzle easily. Think about 1,000-piece puzzle. However, there is no single best. Both of them are still in use in the same rhythm, for example. Why is that? This is short, 50 to 300, can go up to 500 characters, but has very low sequencing error. So the sequencing machine doing really good job at this stage. For the long read, they are really long, can go up to 2 million characters, but we have around 15% sequencing error. So you are not sure whether part of the read is correct or just sequencing error. That's why you need high coverage coming from this, meaning that you need a lot of reads to do overlaps, and based on the overlaps, you can have more confident about each base of these reads. So though it's long, but this doesn't help you to solve read mapping problem, for example, because you need still high coverage with these reads. Okay, so in short, for genome analysis, you already have the chemical format, and you'd like to have it as text format. So you send it to the sequencing machine, you get those bunch of pieces, and then you have the reference genome that we built in 2003 or later updated version of it. Then you pick one of these pieces and try to stick it to somewhere in this reference genome. So you go to the first place, you check whether it's similar or not, then you move on to the next, and so on until you reach the end. And you keep doing this for all these pieces until you get a very nice, smooth, full sequence of our genome. However, when you do city-scale microbiome profiling, for example, where you're looking inside the crowded places, you don't expect a single sample or a single DNA molecule to be in your sample. When you're sequencing it, you will expect you have bacteria, viruses, could be a human genome there, and many others. So when you get a bunch of reads, you can't compare it to a single reference. What you need is a reference database, where it includes all the things that you are interested in or that you are expecting in your sample. And then you start doing the same step as the genome analysis, but the problem, you will get some shared reads between all of these references, and you need to do something about it to really know whether this sample contains this reference or that reference. And for that, we have a coverage threshold. For example, if I'm covering all of it, then I'm having high confidence that this reference or this species exists in my sample. So you can think about it as a collection of genome, which is metagenomics. If you are targeting single genome in a clean environment, then you are targeting genomics study. Okay, what are the challenges in doing such step in read mapping? So the first challenge is you need to find many mapping of each read. Although you check location by location in the reference genome for each read, so you can't stop after you find the best place or the best location in the reference for that read, where that read maps to the reference genome. You need to report all of them. If the same read mapped to this location and many other locations, you need to report all of them because you are not sure which one is the best for you. Although they might have exactly the same similarity, you need to keep all of them. You need to tolerate some small variation or errors in each read. Why is that? Because this is actually the goal why we are reading our genome. We are reading our genome to find differences between the same species or the same or different species, right? We are looking for these variations, so we need to maintain them. It's not enough just to compare the read to a location, the reference with exact match. You need to allow for insertion, deletions, and even substitutions, as I showed in some diseases where you have a large duplication or large deletion of your genome. And you need to map each read very fast to the reference. Why is that? Because sometimes, depends on the application, it could be a life critical. For example, critically ill children hospitalized, you can't wait weeks to do this step. You need to do it in a very fast way. And at the end, after you map all these reads to your reference genome, you'll get something like that. Okay, so we explained about read mapping, but how we perform read mapping actually. So the brute force algorithm is just pick the read, and you have the reference genome there, and go scan it linearly. Location by location, you keep checking, looking for that similarity. However, this is extremely expensive. Why is that? Because at each location, when you compare the read with this portion of the reference, you are using dynamic programming algorithm, which runs in quadratic time. And you need to do this for many reads, let's say the K reads, and you have n long reference. So that will give you this runtime, or this time complexity, which is really expensive. You can't afford it in today's CPUs. So what we do normally is we follow the yellow pages method. So whoever was using this yellow pages in the past to find the phone number of your friend or colleague. So I was using this in the past. I don't know if these days we are still using these in Japan or not. Okay, but for those who knows how to find the phone number of your friend, for example, what you do is you go to the first few pages where you have the index of this book, and you go search for the family name that starts with a certain alphabet that you are targeting. It would be A, for example. Then the index will tell you go to page X, where it contains all the family names starting with this alphabet. Then you go to the step 2, where you retrieve that page X, and you go to that section, and then you already know the first alphabet was A, that's why you come to this section. Then now you check the second character, the third character, the fourth, all the way until you find the full name in that section. And when you find the full name, you just get the phone number of that person. And we do exactly the same way in genome analysis, with no differences. So the first way we need, or the first step, we need to have the index of the book, right? So that's why we build the index of the reference genome. So how we build it, we go to the reference genome, we pick the first piece of the reference genome, we record it somewhere. Let it be 12 long, 12 here, or the K here is 12. We call it K or a seed. And this number doesn't have to be 12. It can be anything starting from 6 or around to 10, all the way to 35, 45. Depends on what application you're using. And then whenever we find the same seed along the entire reference, we record all the location. So this is the start location of this seed. So it appears at 12 all the way to 23. 12 characters. And it appears at this location, that. And we keep doing this for every 12 characters in the reference genome. We record all the location, and this is what we call it the index. Or here, in this example, we use hash table. It could be any index data structure. After we build the index, now we will move to the second step. However, the index, for example, is built only once for each reference. This is the good thing, because this is very time consuming. So it depends on the way you pick these seeds. It could be overlapping, non-overlapping, adjacent, non-adjacent, spaced, minimizers, or even compressed. And depends on the way you pick these seeds, the size of the index really varies. So it could be 16 gigabyte for a human genome, and it could be as well 4 gigabyte. And this will affect your indexing time. It can be just a few minutes, 3 minutes as Minimap 2, or it can go all the way to 1 hour, for example, for other tools. And it doesn't matter which way you pick, it doesn't matter which seed you pick, it doesn't matter what size you affect here, as long as you maintain high sensitivity and you are able to report all the needed location with a correct alignment. So there are many tools using many heuristics to find the optimal seed, for example, to reduce the runtime or to reduce the memory footprint. So for some application, it's okay to have a huge amount of memory here as long as this provides you with high speed up, or vice versa. It depends on what application you are targeting. Don't think about the current conventional computer architecture for CPUs. It could be beneficial for GPU to have a large amount of memory if that helps you to do some kind of parallelism where you divide the index into chunks and then you process each chunk separately. So by the way, each record here in the hash table is totally not related to any record or any entry in the hash table. So you could really divide it into pieces and process each piece separately. Okay, so in step 2, we already built the index, which we built it only once for that reference genome. And we have the reference genome, we got the read. So what we do with the read, we pack the seed the same way as we use for building the index. And then we send the seed to the hash table, we query this seed, and then the hash table will tell us all the locations that this seed extracted from the read appears in the reference genome. Then we go to all these locations, the reference genome, and we extract the segments equivalent to the length of the read or plus some threshold, and then we try to verify these locations in the reference genome, whether they are similar within a certain threshold. As I mentioned before, we are not looking for exact match always. We need to maintain some insertion, deletion, substitution. That's why we need to do some alignment or dynamic programming algorithm where we verify the similarity between these segments. And we don't expect to have a high similarity or a large number of locations where it really fits within a certain threshold. So most of them will be a mismatch, for example. And for step 3, so once we retrieve that section from the yellow pages and we find the name, now we are trying to compare the target name with the name mentioned in the book. So you go to second character, third character, fourth character, all the way until you complete the complete name, then you are sure that this is similar to the name you are looking for. And for that, we are using read alignment step, or verification, or pairwise alignment. Many researchers in this area refer to it in different ways. So, how many of you are aware about edit distance, Hamming distance, or any string similarity method? Okay, not much. Alright, so edit distance is a metric used to tell you how many, or the minimum number of deletion, insertion, substitution. So the less is the best for you. It's closely similar, let's say, about it. So for example, organization and operation. We want to know the edit distance, how similar they are to each other. So if we compare them, we got 2 insertion here and 5 deletions from here, for example. So what will be the edit distance? 7 operation, because we perform 5 here and 2 here. So this is similar to this within an edit distance of 7. So if your threshold is 3, then those are not similar. If your threshold is 10, they are similar. This is how we define similarity in genome analysis. So similarity here doesn't mean to be identical. And there are many ways to align this to that. You can see here there are 2 ways, for example. Organization and translation, same thing. You can find 3 ways, for example. However, the distance is 4, not 5 as here. So this is way larger than this, for example. So we don't pick this as a distance, because by definition it should be the minimum number of operation. We can use other metrics, the way you like. Instead of edit distance, you can use Hamming distance, which is only substitutions. rather than insertion and deletion. And that really depends on what you are looking for. If you are targeting certain disease that's caused by single snap, then it doesn't look wise to use edit distance, for example, because you need single snap. So Hamming distance can provide this information for you. And there are read alignment tools using only Hamming distance, such as MISFAST Ultra, and so on. And there are other tools as well using the edit distance. So both of them are still used until today. Am I going fast? Good? Slow? Okay. All right. So now we finished step 2, step 1, 2, and 3, and doing read mapping. And this is one of the alignments we published in 2009, where we have a hash table based. And we built it as a comprehensive tool where we report all kinds of seeds exist in the reference genome. It's not minimizer, there's no heuristic to reduce the workload. We just pick all overlapping seeds. That's why the size of the index is really huge, 15 gigabyte. But this is beneficial for some of the application, and for some other applications, not good. And when we analyze the latest sequencing machine, we find that, as I presented before, that it can produce around more than 300 million bases per minute. And when we go to the read mapping tool, the latest one, then you can find it's only able to process 2 million bases per minute. So you can see there is a bottleneck between the sequencing machine and the read mapping. And this number is not necessarily correct. It could be way more than this. We have 150x slower between this process. So if you want to cope with the workload you are getting from the sequencing machine, you need to accelerate read mapping process. Okay, so before accelerating any tool in the world, not related to bioinformatics, could be anything in computer science, you need to know the causes for this slowness. What caused this read mapper to be slow? So when we did some analysis, we find that more than 93% of the time spent in read mapping is spent on dynamic programming algorithm, which is the verification step, or calculating the edit distance. Of course, this depends on the tool you are using. Some of the tools are using more heuristics than the dynamic programming, so this percentage can go down to 50% or 60%, but still more than half or 2/3 of the runtime is spent on the dynamic programming side.
Can I have a question?
Yes.
So it's kind of surprising that this bottleneck was not revealed until 2017, right? Do you have any comments on that?
So basically, they could have it. They could have the exact same observation. That's why we have a lot of acceleration work in the past, even 15, 20 years before that. But maybe they don't have the numbers, for example, or it depends on the tool you are analyzing, or they don't report it for some reason. I don't know, but the observation is still there. That's why we have all those acceleration work in the past. Any questions? Okay. So, yeah. This is Mr. Fast, which is the one I present. Yeah, Mr. Fast in 2009. This paper. This paper got updated in 2013 with a different version, different optimization. They call it Fast Hash, or Mr. Fast with Fast Hash. But the same tool. So when you go to this GitHub page, you'll get the 2013 version, not this version. But we analyzed BWA map, Minimap 2, all of them have more or less the same percentage here. It's around 50 to 90%. It really varies, depends on the heuristics. 80 to 85. Okay, yeah, somehow close to this. And it really depends on what algorithm you are using. So when you have the hash table or the index data structure and you want to query a seed there, so you get many locations, right? Then you perform dynamic programming on all of them or some of them. This is what affects the running time. So if you really would like to go with the best way and perform dynamic programming with all of these locations, then you'll get bottleneck really high. But if you use some heuristics to reduce that workload and apply dynamic programming to only a few of them, then the percentage will get reduced here. But still, you are bounded by dynamic programming algorithm. Okay, the second observation here, whenever you go to the location and you verify it using dynamic programming algorithm, you will find 98% of these locations having higher dissimilarity more than the threshold. So if you are interested in only 3 differences between the read and the segment of the reference, you will find most of these locations having higher than your threshold, meaning that you are wasting time on verifying something that you don't really need. The third observation. Okay, so dynamic programming is expensive. Why we are using it? Why not finding just linear time algorithm and just start using it? Okay, so the goal behind using dynamic programming is that we really need to enumerate all possible prefixes between 2 strings. And for example, Netherlands and Switzerland, you want to compare these 2 words. So what you do is, having all enumeration between Netherlands and Switzerland, you start with Netherlands with S, Netherlands with SW, all the way until you reach Switzerland. So you can see a lot of repeated computations that is involved in this comparison. So the best way to go is dynamic programming. Why is that? Because you store some of these computations in the cell and you revisit them later on when you need it. So in this way you save computation. We claim that we save computation by dynamic programming, but the way we going back and forth, revisit these cells and move on, these will create a lot of data dependencies. So these data dependencies limit you, or the way you are computing this dynamic programming table. So you can't do anything to accelerate it, because you really need to compute the entire row, then the second row, then the third row. You can't compute the second row before the first. But you can compute all these cells in the same row in a parallel fashion, because they are independent, or they can be dependencies as well. So you can either compute the rows or the columns, but there are a lot of data dependencies, because at each cell you need to have this cell, and this cell previously computed before you can compute this cell. So you either can go rows or columns or anti-diagonal shape, for example. You can go tiling and all those other heuristic methods to perform dynamic programming, but the data dependency is still there. So if you want to accelerate it in FPGA, GPU, you can't do anything about it, unless you can place some large number of cores where each core performs single dynamic programming problem. So you can solve it for many texts in parallel, for example. And the third observation, for example, if you want to calculate a global alignment. So you can't do anything until you compute the entire table where you reach this cell. And this cell will tell you that you have 5 differences at this location. So you can't stop in the middle if you're exceeding a threshold. You really need to compute all the way until you reach the end here, and then find it. So this is very expensive to compute the entire thing for 2 strings that do not match together, for example. But if you are using, for example, a local alignment, it will be way easier for you. You don't need to wait until the end, for example. Okay, so now we show the challenges, we show the observation, why it's slow, and now we are going to present some of the algorithms used to accelerate this. Do we have any questions before we proceed? Okay. So our goal in this work, if anybody watched these 2 movies, Gattaca and Tomorrowland in 2015, recent one. So they have this smartwatch where they put their finger and the watch will pick, using microneedle, a sample of your blood and analyze it, and they recognize your identity based on your DNA in just mere seconds. So it's really in the same moment. So we would like to have something like this for genome analysis. And if we have this device, for example, it will be really great, really helpful. And this is true for all other areas. So I'm going to show you examples where in machine learning, artificial networks, even autonomous driving cars, they're really going forward with these devices. not necessarily to analyze DNA, but to do something specialized for other applications. So our goal here is to accelerate or reduce the time spent on calculating the optimal alignment in genome analysis using new algorithm and hardware accelerators given limited computational resources. So of course we are not looking for a cluster here. So we could easily place many cores, thousands of them, millions of them, and aim that we are accelerating genome analysis. No, this is not the way to go. We need to do this in single CPU or single FPGA board, single GPU, and so on. So for that we have an open question. Until now, nobody can address it fully. So we don't know how and where to enable fast, accurate, cheap, privacy-preserving, and exabyte scale analysis of genomic data. So we really don't know where to perform this analysis. We have too many options. Not necessarily in CPU, we have way too many hardware acceleration platforms to do this. So one way to go is with conventional computer architecture. You have the storage, you have the main memory, and you have the CPU. And this is the sequencing machine providing you data. Would you like to wait 44 hours until you get all the reads or the FASTQ file from the sequencing machine? Then you store it in your hard drive, then you move it to the main memory as full file or chunk by chunk, and then process in the CPU, then store back the result to the storage. This is very energy inefficient, very time consuming, and the way to go is pushing towards a new architecture customized for genome analysis. So we are very good at building supercomputing architecture or nice computer architecture, but we really don't know how to do with genome analysis. So we want something that we process genomic data where it makes sense. If the data is here, why do we need to move it to the storage, then the main memory, then the CPU? Why don't you do processing inside the sequencing machine, for example, or directly leverage FPGAs or GPUs, or even all of them as heterogeneous architecture, where you can provide some SIMD instruction to accelerate the dynamic programming part, but for the hash table you need a huge amount of memory, so you can process it inside the storage, for example, if you are able to do that inside SSDs, for example. So we really know that most speed up coming for any application should come from the parallelism enabled by novel architecture and algorithms. We can't just design new algorithms for current CPU. We can't just design new architecture for existing algorithm. We really need both directions to go with high speed up. And this is true for many applications. For example, this is one of them, Preferred Networks, a Japanese company, where they are having this MN core in last year. They have 4 dies, for example. This is specialized for accelerating deep learning network. And they have some matrix arithmetic blocks in each of these pieces where they can perform many computations in parallel at the same time in a dedicated chip. We have many examples of this. Most of them are recent in these years. For example, Surprise wafer scale engine this year, which is way larger than largest GPUs we have. So think about this, such customized chip that can process your genome in a few seconds. And this is built for accelerating machine learning software. And this is a French startup called UPMem, where they enable processing inside DRAM chip. So this is a DRAM chip, normal one. You can plug it in your computer, and it acts exactly as a DRAM. So you can store, you can retrieve data, all of that. But it has processing cores inside the DRAM. So you don't need to move the data outside the chip itself to the CPU, for example. You can do some simple bitwise operation or somehow complicated operation inside the DRAM. So that will save you a lot, especially for energy-wise, because you don't need to transfer the data through power-hungry buses from the DRAM to the CPU. And another example, or last example, is Tesla. They have their own dedicated board to do some processing. They even have some duplicated chip. So this is exactly the same chip In case of failure, one of them, the other one will operate normally. So this is for reliability. They have even duplicated camera, power supplies, and so on. So we really need a customized solution for genome analysis because we have a huge amount of data and a huge need to process this as fast as we can. So this is another good news happening in 2018 from Illumina. Illumina, which is a company producing those very short pieces of your genome. They acquired Genome, which is another company or a startup building or leveraging FPGA board to do some processing for genome analysis. So you can think about the future where Illumina can do some processing using FPGA board inside their machine, rather than moving the data to your CPU and do some processing there. This is one of the benefits. The other benefit, think about you do processing in real time. Because I told you that this machine produced some data in 44 hours. But this is not always the case. You don't have to wait 44 hours to get the data. So the data will be streamed in real time. So every few seconds or every few minutes, you will get a chunk of this data. So if you are able to process it as it gets produced, then you can do real-time processing in a very fast way, chunk by chunk. You don't need to wait 44 hours. And at the end of the sequencing, you will be already processing all of these data. Another news, so this is Illumina, the short-read company, and this is PacBio, another company producing much longer reads than the one produced by Illumina. And there are a deal negotiation going on that Illumina will going to acquire PacBio. Still, the deal is not set yet, but this has been in the news for some time. So let's see the future of read mapping or read sequencing, how it will be in the next 10, 20 years. Maybe we'll have a single machine that produces way longer and accurate reads using the same machine, and you have some ability to do processing in this machine as well. Okay, so now we know that it's slow. We really need for some specialization to accelerate the read mapping process. So what are the direction? What are the other researchers doing to accelerate this problem? I'm not sure if we should stop here, take a break, or continue for 10 minutes.
Okay, then let's take a 10 minutes break from now.
Okay. So we'll come back after the break. So now let's continue with the ongoing directions to address the slowness of read mapping process. So there are three main directions. The first one is seed filtering technique. So as I told you, there was an index where you query a seed to that index and give you many locations. So there are some methods, some heuristics, some new algorithms to reduce the number of these retrieved seed from the index. And the second direction is to do pre-alignment filtering technique. Pre-alignment, so as you can see from the name, pre means before the alignment. So before performing the dynamic programming, you do some filtering such that you reduce the number of locations for the segments to be verified. And the third direction is to accelerate the dynamic programming itself. Though, as I told you, there are data dependencies, so you can't do much about parallelizing the algorithm. But I'm going to show you some of the work that can compromise the accuracy and gain some speed up. Okay, so this is an example using the previous example, but now showing you the ongoing directions. So you got the seed from the hash table, you got the location, but when you go to the reference genome, you don't retrieve all of them. So you try to reduce them, then you get some of the segments, not all of them. Then you apply pre-alignment filtering, not the dynamic programming. Then you apply the dynamic programming only if it is a perfect match, or meaning that a match within a certain threshold. And here, if the pre-alignment is not showing that it's not a good match, then you ignore the dynamic programming part. Okay, so now let's see the seed filtering. So this is one of the work we published in 2013, which has two key ideas. So the first observation is that we are looking always for adjacent k-mers. Adjacent k-mers meaning that if they are adjacent to each other in the read, then they should be adjacent in the reference genome as well. And I'm going to show you an example here. If this is the read, and we are picking 3 seeds, non-overlapping seeds. So we have the A's, the C's, and the T's. And when we query the hash table, we got these locations. So if the read is next to this, if the seed next to this, and this next to T's, for example, they should be located next to each other in the reference, for example. So apparently this is a good alignment. This is a good location to be aligned. But all of these are really far, unless your threshold is really high, so you can consider some of these locations. So this is a filter, we call it adjacent k-mers, which reducing the location of retrieve from the hash table, for example. So the second contribution of this paper is called cheap k-mers. So what does that mean? So I'm going to show you in the example. This is the hash table where you got some of the location retrieved from the hash table. So here are retrieved only 4 locations for some seeds in this reference. And for many of these seeds, you got different list of locations. So you don't expect them to be equivalent or the same number of entries here. And you set a certain threshold, let it be 500, for example. So whenever your list exceeds this threshold, so ignore that list totally or ignore whatever exceeds that number. So you can consider here the first 500 and then ignore the list of the location, the rest of the locations, or you can totally ignore this list, and that list. And you only consider these lists because you consider them cheap to be verified or computationally cheap compared to other, compared to verifying all of these locations. Now, what's the insight or the theory behind this cheap k-mer? So basically, they said whenever you have a large number of locations, probably this seed is, let's say, false positive. So it's coming from repetitive regions, for example. As we know, the human genome has around 50% of it, just repetitive regions. So if you have a read that mapped to too many locations, then probably this is not a good match. So probably we are looking only for the few locations. Again, this is a heuristic, doesn't have to be true. But it's proven that when you use this method, it will give you the same sensitivity as you are verifying all of these locations, but with much speed up, with less execution time. Again, how to set the threshold, it's not known. This is set by the user. So you can see there are a huge reduction in the location to be verified, for example. And this is what we call a fast hash paper. So I'm going to skip this. Yeah, this is the paper. You can see the source code is here, published online, so you can play with it. You can try different thresholds, different ideas. For example, if I would like to have a different threshold, different strategy, your own idea, you can optimize this algorithm, for example. Okay, now the second direction is to do pre-alignment filtering. This is the direction that I like myself, and where you can contribute really to accelerate the entire pipeline, for example. So we know that dynamic programming is expensive, and our goal is to reduce the need for dynamic programming. All of us agree that we cannot ignore dynamic programming algorithm. We should use it, because it's the most accurate algorithm that can provide you the result that we are looking for. So what we can do is just to have an algorithm that can tell you in a very fast way whether these two strains match within a certain threshold or not. We don't care about number of differences between these two strains, just we need to know whether the differences exceed or do not exceed the threshold. So this is our method, reducing the workload coming from the hash table all the way to the dynamic programming algorithm. So you can have many of these pre-alignment filtering next to each other. One of them, for example, this is very fast, but having a lot of false positives. So you send the correct one with the false positive ones. And the next stage, which is a bit slower but more accurate, then until to the reaching the last stage, for example, which is the most restricted stage, for example. However, for these, we need to be careful because this is a filter or a step coming before the dynamic programming. So we need to filter out most of incorrect mapping. All of these reds, we need to get rid of these locations. But at the same time, we need to keep all the correct ones. We need to preserve all the correct mapping. If you are removing some of these correct mappings, then we are producing false negative. We don't want false negative. We want false positive. It's okay to have it. Why is that? Because anyway, we have a strict stage that can 100% verify whether this is a good match or not. So as long as you are not substituting or you are not replacing the dynamic programming algorithm, it's okay to have false positive, but it's not okay to have false negative. Meaning that the correct, keep it, and the wrong one, remove it. This is what we do. And of course, we need to do this very quickly, and this is the goal, actually, of having this filter or this step. If this is slow, then why we add a new step here? We could keep the dynamic programming algorithm, for example. So the first pre-alignment filter called gatekeeper. So this is our work published in 2017, and I'm going to present it now. Okay, the key observation of this work: if you have a two-string, differ by E differences, could be insertion, deletion, or substitution, for example. And every character or every base pair match can be aligned in at most 2 E shifts. Now I'm going to show you an example of how we can perform this matching between the two strings. And the key idea of this work is to compute shifted Hamming distance and then perform AND operation of all of these masks or all of these vectors, and then identify invalid mapping. And because we use just bitwise operation here, then this sounds ideal for FPGA platform. The key result gatekeeper is 90x to 130x faster than SHD, which is a previous pre-alignment filtering, and better than the adjacency filter, the one I presented to you, which we reduce the seed based on their location next to each other. And of course, we have false positive coming from this filter. And when we add gatekeeper as an FPGA to Mr. Fast, which is a mapper, a CPU mapper, we achieve 10x end-to-end speed up and remapping. Okay, so how we perform all of these factors. So all of you know Hamming distance. Hamming distance meaning just performing XOR operation between the two strings. So we have two words, Istanbul, Istanbul. We want to compare them using Hamming distance. So just perform XOR operation between them. And you got 8 matches because they are perfectly identical to each other. Now, I want to do an operation myself. So I want to delete something. Let's delete the character A, for example, from here. Now, whenever we delete a character, all these trailing characters will be shifted to the left, as we can see now. So now if I want to compare again the two words with the modification I did, if I do just Hamming distance, then I will get 5 mismatches rather than just 1. Although I just deleted single character, not 5. So how to fix that? Because when I delete one character from here, all these trailing characters get shifted to the left. So I'm going to have another copy without this shifting affecting the word. So I have this now the same 2 words with the deleted character from here. And now I'm having another copy of it, but shifted by 1 step to the right. Now if we do the comparison again between the 2 words, so here it sounds like 3 characters exactly perfectly matching, and another 4 characters from here exactly matching, right? So if I do the XOR between the 2 words, I'll get this result. So now what I'm looking for is I want these 4 zeros and the 3 zeros, right? Because these give me that this is a perfect match here. So how to collect this result from the 2 vectors? So the way to go, maybe to do an AND operation between these 2. So if we do AND operation, the 0 always dominate. So I will get 4 zeros, 1, and 3 zeros. So this vector is telling me that there is a single mismatch here, somewhere in the middle, right? So by doing this, I already fixed the problem with doing edit distance calculation correctly. Do we have any question here? Should I repeat it? Is it clear? Okay. So it's clear, right? Should I repeat? Okay, okay, good. All right. So now we got this result, and this representing that the zeros are the matches, the one as the mismatches. So it's telling me that you already did an operation at this location, for example, which is A, so which is true. So now I already did myself the deletion, but in fact, we don't know whether it was a deletion, insertion, or substitution. So I can't assume anything about the alignment. So what we do is more general solution than this, is I'm having the query and the reference, which are two segments, and I will build all of these vectors. What are these vectors? So these are 7 many vectors based on a threshold. So the number is, for me, is 2e plus 1. So what is E? E is the distance threshold. So I'm setting the threshold here to 3, which gives me the result of 7. That's why I have 7 vectors here. So what are these 7 vectors? So the first vector is just XOR between the two strings, which is Hamming distance, basically, or the Hamming mask. And the next 3 vectors, because I don't know whether they are deletion or insertion, so here I'm considering as deletion. Deletion, when I delete a character, then all the trailing will be shifted to left, right? So I should have this vector to be shifting the query to the right by 1 step, then do XOR with the reference. So I'm shifting this guy 1 step to the right, then do XOR comparison with the shifted version with the reference. This is how I get this second vector. How I get the third vector, so I'm shifting this by 2 steps to the right, same direction, then compare with the reference. And I'll keep doing this, gradually shifting and XOR until I reach my threshold, which is 3. So I'm getting 3 masks that I'm calling them deletion. Why deletion again? Because when I delete a character, I'm shifting all the trailing to the left. So I want to cancel this effect by doing the opposite. So that's why I'm shifting to the right, not to the left. By 1 step, then compare. By 2 steps, then compare. By 3 steps, and compare. And I'm doing the same thing, but the opposite direction with the other last 3 vectors. So how I generate this vector, I'm shifting this to the left, not to the right. So in this deletion mask, I'm shifting to the right. Now here I'm shifting to the left by 1 step, compare, shift 2 steps to the left, then compare. And this is how I get the result of XOR operation for all of this. Is it clear? Yeah, sorry, the example has a lot of zeros, a lot of characters, so you may not be able to track the result. But you can see maybe from here we have 4 A's, 4 A's. So if we shift it a little bit to the right, then this A will be aligned with this A. That's why we get a lot of zeros here. Because this apparently the query is shifted to the left. Even here you can see it, 4 A's are shifted 1 step. That's why we get a lot of zeros in the second vector. So this is the solution for you. This is what you are looking for after you're doing all of this. You want really to track these green segments. And these green segments will give you the correct answer about the number of differences or the similarity between the 2 strings. And this is our goal, actually. We want to find any mechanism, any way to track these green with the correct results. And how to do that? So you can think about first, before thinking about how to collect these green zeros, you can think about having these 2e plus 1 vectors. So now we are able to have any type of deletion, insertion, substitution, any combination of these variation, right? Think about having 3 deletion, then followed by 3 insertion, and then some substitution, and so on. So this is a global solution, right? We can provide you optimal result, or not optimal, sorry, because this is heuristic, this is a filter. So it shouldn't provide you optimal, but can accommodate many number of variation specified by this threshold. So now one way to choose, as I presented in the previous example, is to perform AND operation between all of these masks together. So just AND them together. When you AND these vertically, So whenever you have a 0, the 0 will dominate, right? So it will cancel everything else. So if you have a 0 here, the result should be 0, right? So let's get the result. Yeah. So this is the result of having the XOR operation. However, when you do the AND operation, we are looking to have these 2 1s, right? Because this is part of the solution. Having 2 1s meaning that there are 2 mismatches between the long matches. So if we got this in the result here, and we got this one here, and this one here, and then at the end we count the number of 1s, then we'll find there are 4 mismatches, right? But when we do AND operation, think about the 0 here, and we have 1 here. When we do AND operation, what will happen to the 1? It will be cancelled, right? Because 0 always dominates 1 with AND operation. So that will cause you a false positive, right? Because when you reduce the number of 1s, you are reducing the number of differences. So if your threshold is 3 and the differences are 5, but when you do AND operation, your differences get reduced to 2, for example, then you are not exceeding the threshold, then you allow those 2 strings to be sent to the dynamic programming part. To fix that, we try some heuristics. We said, okay, look at the vectors. What pattern do you see? There are a lot of single 0 and 2 zeros everywhere because of the random shift at the comparison. So we randomly shift 1 step, 2 step, 3 step. There's no reason why we keep shifting all of those gradually. So they don't have any meaning, basically, these random zeros. So we said, okay, let's flip them. So we convert these vectors, all of them, to these vectors. You can see this is somehow clean. We have less number of zeros, especially the single 0 and the 2 zeros. They are already cancelled. So the remaining are 3 zeros, for example, or very large number of zeros. Now when we do the AND operation, we got some of the results are correct. And we still have a problem, for example, with this one, because we have a 0 here, which is a part of large number of zeros. So the number of 1s here get reduced by 1. Again, we have false positive, but it's still okay, as long as you don't have false negative. And you can see the reason why we don't have false negative at all. Because we are always underestimating the number of 1s. We never overestimate it. And this is the correct result coming from, for example, Needleman-Wunsch or global alignment algorithm. So you can see you got the result exactly the same position as a global alignment algorithm. So you might use some heuristic to leverage this information to give you the cigar string without the need for dynamic programming algorithm, for example, if you find a way to find these locations correctly. So you can think about, you have already substitution here and deletion here and so on. How do you know that this is a deletion? Because this exact match coming from this mask, which I called it previously first deletion mask, right? So I know if an exact match coming at this mask, meaning that there was a deletion before. That's why I get the matches at this mask. If the matches are at this mask, then I shouldn't get any deletions, right? So you can think about any improving or any future work to improve this work and producing cigar string, for example, or perfect alignment out of this filter without performing dynamic programming. But you should be careful about that. The cigar string should exactly match the one provided by accurate algorithms. Okay, so what we... Yes?
How is it false positive zeros connected? Is true ones still over to the next terms or something else?
The false positive.
How to correct it? How to change zeros sandwiches by true ones to correct ones?
Okay, so how we flip the zeros into ones.
Yeah.
Okay, so I didn't tell you something about the result here. So in the final result, we should be careful that we flip the 0 into 1s, right? So when we flip the 0 into 1s, you might be flipping a correct match. There might be here, for example, think about having here, this green match, having this pattern. Here, think about having it here, this correct answer. If you have this and you convert it into something like this, and you get this pattern in the final result instead of this, what will happen? So you will get 3 mismatches instead of 2 mismatches. So in this case, you are overestimating the number of matches. So you'll get false negative, right? So to do this calculation correctly, you need to do the opposite operation, the one you did in these factors. So when you convert this pattern to this, here, these factors, so you need to do the opposite when you calculate the final result. So in the final result, when you get this, you go and find whenever you have 3 1s or 4 1s. So 3 1s, probably you are not sure if it's coming from correct result or from flipped one that you already flipped just a step before, right? So to be careful and to be on the safe side, you always consider that these coming from the flipped one, flipped 0. So you change them back to 1, 0, 1, and you change these back to 1, 0, 0, 1. And then you calculate the number of ones. You do this step only for this vector, but you do this step for all these vectors, just to be on the safe side. And how you flip these zeros. So what we did, because we implement this in hash table, so you consider every 4 bits. You go through the vector and you scan every 4 bits. And whenever you see this pattern or this one, then in the lookup table itself, you just flip it.
Is the 4 bits overlapping?
Sorry?
Is each 4 bit overlapping or not?
No, shouldn't be.
Distinct.
Yes.
So,
If you overlapped it, then you might repeat yourself, right? You might repeat the same computation. Think about having this, for example, and you want to convert it.
second one is
So now, if you take 4 bits here, and then you fix this, because you find 1, 0, 1, then it will be 1, 1, 1, right? But you will have 0, 1. Now you already finished calculating those, right? But you didn't fix this pattern. So how you fix it?
I wonder if it's possible to fix that 0 by shifting the window to right by 1.
So, okay. So the first, maybe I can show you now. So the first, there are 2 things. How to fix single 0 and 2 zeros. This is one issue, how you can fix it. And the second thing, how you fix this kind of pattern, for example. And for us in FPGA, we do it as overlapping lookup table processing to find of 5 input, not 4 input. Why 5 input? Because we are looking for at most this pattern. This is the hardest one. This is easy to fix, but this is the hardest one. And we have 0, 0, 1, and it might be something before it or after it. So we don't know if we will get this kind of pattern because we have a 5 input lookup table, or we will get this pattern. So to fix these 2 patterns, we need to consider 5 input lookup table. And we do it in overlapping fashion that I'm going to show next. And these lookup tables are independent of each other. So you can solve all of them at the same time.
On FPGA.
Yes. Yeah, it will be like 3 slides next. So I'm going to show you that later on. Okay? All right. Any question? Okay. So, by doing that, all those bit vectors operation, we are resolving the data dependencies, the one we see, the one we face in dynamic programming algorithm. So instead of having all of these data dependencies calculations, regardless of the algorithm we are using, global or local, Needleman, Moore, Smith, Waterman, What we are doing just individual comparison between those characters. So we just compare the A with C, with T, with A, and we have individual result. If it is a match 0, if it is a mismatch 1, and that's it. We keep doing this for all those characters. And this is the one we generate using just the XOR operation. And this is the one we use to generate by shifting by 1 step then XOR. And this is shifting 2 steps and then XOR. Then this is shifting 1 step but the opposite direction, then XOR. And this is shifting 2 steps and then XOR. So this is exactly as the example you saw before, but it's laid in diagonal shape. Any question? So you can see there is no data to be density here at all. You can compute everything in parallel, everything at scale. So these cells within a diagonal are independent, and these factors between themselves are independent. So your goal is always to track these zeros. Whatever algorithm you use, you want to track the longest number of zeros and these zeros. And after that, you can use GPU, FPGA, SIMD, whatever acceleration platform that can process bitwise operation, for example. And for those of you wondering, we have SIMD, for example, or multi-core processing. We have GPU. GPU used for image processing or something with floating point operation. It's really preferred for floating point, not fixed point. But the good thing with GPU is that you have a huge amount or a huge number of threads. So you can think about it as multi-threaded program for CPUs. So you can launch all of these threads, but all of them doing the exact same jobs. So you need to divide your computation such that you are doing repeated jobs over many threads, for example. For GPU is a little bit different because it's hardware architecture where you have some units and most of them are lookup table. Lookup table, so lookup table, it means to do some logic operation using AND or XOR operation. But you can implement any algorithm or any function using these logic gates. So by storing these values and retrieve them back from lookup table, you can perform any operation, right? You can use even the memory of the GPU, the FPGA, to store the result of some very complicated operation, then retrieve it back if you have a certain pattern in your algorithm. And for FPGA, the hardest part is to do some reconfigurable parameters. Think about the edit distance threshold, for example. The user can set it to 2, but later on, next step or the next experiment can set it to 10 or 20 or 15, right? FPGA doesn't provide such flexibility with programmability. So what you need to do is redesign your hardware architecture totally, maybe, or do some changes to your hardware, and then move the code or the RTL to the FPGA, then run the FPGA again. But for GPU, it's way simpler because just as a CPU program, where you change some parameters in the program and compile it and run it again in the GPU, as easy as this. Or you can do some for loop to have a variable as changes from 0 to 100, for example. So repeat all the experiments from 0 to 100. You can do this simply as a for loop, but in FPGA you can't do for loop, for example, because it's hardware. You can't think about it as a software. In hardware, you can't say, I want to repeat this experiment this many times. You can't do that. You really need to design the experiment itself inside the hardware chip, where you have many lookup tables. So what you can do is to determine the number of the lookup tables that you are going to use. So saying that I'm going to use 5 lookup tables, for example. That's it, done. You can't change this later on to 6 unless you redesign your code, your RTL, and so on. So I don't have time to explain more about the FPGA, but if you have any question, feel free to ask.
So do you have any theoretical analysis about the false positive rate?
Yes.
That algorithm?
We did some analysis in a paper called Gatekeeper. I'm not going to present it here because it's more like what are the sources of the false positive. But it's public online, anyone can access it, called Magnet. So if you search for Magnet, you can find it. We did a comprehensive analysis of all type of false positive we can get from Gatekeeper, for example, and how to address them. And this was the way for us to go and improve Gatekeeper.
And I've got a question.
Yeah.
What kind of the range of E do you have in mind? I mean, in the long read alignments, we may have a very, very long reads like 1 megabase pair or even more, where we need a bandwidth of a kilobase or even more. But obviously, the bandwidth of 1 kilobase doesn't match to this algorithm. So I'd like to know the range that you assumed in your mind.
That's true. So from algorithm point of view, there's no restriction, right? Because you could generate a million of these factors and then do AND operation. But there is a point to worry about is the false positive. So we find that after edit distance threshold of 8, you no longer can filter anything. You will have 100% false positive.
You mean 8 means 2 by 8 plus 1 equals 17?
Yes.
Oh, okay.
So after generating 17 vectors, you're no longer able to filter anything. Why is that? Because you will have random zeros everywhere. You can't fix them.
Even for practical data, like not random?
Yeah.
Okay.
Because you are doing the shift process at random scale. So you are shifting always by 1 step, 2 steps, 3 steps, 4 steps, regardless of the error. So you could have one deletion, but your edit distance threshold is 17. So you could have an exact match as well, and you will have a lot of vectors for no purpose. This is the problem with Gatekeeper. But later on we fixed that issue in the later stages, and we improved the false positive. We reduced a lot.
Why
So-
it's 8? Sorry.
Yeah.
Why
Yeah,
it's 8?
okay.
My intuition was even if you the E parameter is 1, you may have 100% positive if the data is completely random. No?
No, it's really hard to have that.
Right. For practical data, it should be slower, but
No, the amount of computation you perform is
Uh-huh.
exactly the same regardless of the data. You always generate these factors,
Right,
you always
right.
do AND
but
operation.
the false positive rate
But the
would
false
be
positive rate,
different.
so it could be, really depends on where you get these random zeros and the actual differences. For
Right.
example, you might be lucky to have these random zeros somewhere within these zeros, right? So these zeros will cancel any random ones or zeros, right? It doesn't matter. But you'll be unlucky to get these random zeros within these ones in the same column. So that's what we can't expect,
right.
right? So it really depends on the data, yeah, that's true. So you could get these zeros here within these ones, then you can't do anything about it.
Okay.
So the exact false positive, we cannot expect it.
Yeah,
right?
yeah, yeah.
So your data could be anything. Because we are looking for short reads.
I see.
Why short reads? Because we are restricted, as I said, E equal to 8 is the most. And normally we use the edit distance threshold to set to be 5% of the read length. So if your read length is 100, normally the edit distance threshold used in many of the experiments is 5% of that read length. So if it is 8, then the most you can go 8%. Or 8 with read length of 150 megahertz or 300.
You assume or assume this algorithm is constant length inside the camera much. I mean, if we put maximal exact match, it tends to have a gap right next to the boundary. So, in my opinion, this algorithm very effective
So in this algorithm, we don't consider any seeding, right? So the seeding is already done before, and you say like whatever heuristics you use, your heuristic says, okay, this is a good match, send it to dynamic programming algorithm. But then our step or gatekeeper said, okay, stop. I want to check this location before you proceed to dynamic programming. So I don't leverage any information coming from the hash table. I just leverage the read and the location of the reference. I go to that location of the reference and they pick the same length of the read. Then I have two segments, then I do my verification. But whatever seeding I'm using, where are these seeds, whether exact match, maximum exact match, I don't leverage this information. So it's independent of the seed you use. Okay? And this is probably time inefficient, or is not efficient to not use this seed information, because you already know from the seed that there is exact match here. And again, this is a filter, right? This is not dynamic programming. So you can leverage this information safely, right? By knowing there was an exact match here, exact match, so you don't need to perform AND operation again, because you already know that this seed telling me there's exact match within these 16 characters. But for FPGA, we don't care about this optimization, because at the end we are bounded by the clock cycle. So every clock cycle we are doing a certain amount of jobs, right? So we can't optimize anything if we leverage this information or not. Because in FPGA, it's hardware, all of these lookup tables operate by clock cycle coming from the slice of the FPGA or the flip-flop of the FPGA. So everything is synchronized by a clock cycle. So at the end, what is telling me if this is a good design or not is how many clock cycles you need to perform this operation. If there's one clock cycle, then you are done. You can't do anything about it to reduce it to less. But you can pipeline it and use other optimization methods, for example. But for CPU, it might be efficient to combine seeding with gatekeeper, for example. Any questions? Okay. By doing this, this was the first FPGA work doing this pre-alignment filtering on this platform. According to our knowledge, I couldn't find any work doing FPGA pre-alignment filtering. And the goal was for us to do FPGA filtering is that you have a huge amount of workload. Here you can see, for example, you have a large number of locations to be verified by dynamic programming. So we said, okay, let's use the FPGA because it's very fast. Verify all of these locations, then send only a few of them that are accepted from the filter to the dynamic programming part. And this dynamic programming could be anything. It could be the GPU card you designed before, or the FPGA board, or the CPU itself, or whatever platform implementing this dynamic programming. So if you use the FPGA, it sounds like ideal, because you don't need to transfer the data back from the FPGA board to the CPU. Think about always, data movement is bad. Whenever you transfer data from CPU to FPGA, CPU to GPU, you need to do something to hide the transfer time. Okay, so this is how we implement it. Back to your questions. How we flip these zeros. So we use 5 input lookup table in overlapping fashion. Why 5 again? Because we are looking for this pattern. So it could be 101, but this is more challenging. Anyway, it's already covered inside the same lookup table for 101 or 1001. And it could be here anything. And always we are fixing 1 bit per the lookup table, which is the one in the middle. So the output of, if you can see here, the output of this lookup table would be 1 bit with this one, either flipped or not flipped. That's why we will maintain correctness here. So when you have overlapping, the second lookup table here will fix the second bit, not the first one. And by having overlaps, we can fix this pattern, for example. So one of them, for example, will consider these 5, and the second one will consider these 5. So when I check these, I find this pattern, right? But where is the output of this lookup table is the one in the middle, right? So using this lookup table, I'm not fixing anything because I find this to be 1. So just ignore this lookup table. Now when I move next, I find here 0, 1, 0. So I'm going to fix which one here. Then you can find here it's 1, 0, 1. Then I'm going to fix this into 1, and so on.
Did you intend the size of input or 5 bits is equivalent to the size of native lookup table in FPGA?
Very good question. So in the FPGAs, different specification on the FPGA is based on the number of input for that lookup table. So the most recent one can go 5 to 6 or 7 inputs. This is the latest one. And the oldest one can go to 2 input or 3 inputs. And which design choice you should pick? The FPGA that provide you 6 input lookup table or 2 lookup table, 2 input lookup table. So it's always it's better to design your algorithm based on the hardware you have. That's why I mentioned that earlier in my talk, that you don't need to only implement existing algorithm in FPGA. You really need to tweak it to the hardware you have in hand, such that you perform higher performance. So the ideal way is to use single lookup table, right? So what is the single lookup table we have in the FPGA we used was Xilinx Vertex 7 VC709. It has 6 or 5 input, depends on the number of outputs lookup table. So this was our design choice, right? So we choose 5 input lookup table because the FPGA have now. This is a very good question. And that shows you the really importance of knowing the hardware in hand. It's not enough to get an online course or learn from your course how to use FPGA and then implement your algorithm. You really need to know everything about the hardware you have, what affects your performance, what are the time latency, the power efficiency, which design choice I should choose, which one is better for me, which one is better for the algorithm, and so on. And if you find any difficulties, you could tweak the algorithm, change it, and then design the hardware for that algorithm. So everything sounds like very perfect for FPGA. Everything is bitwise operation. It sounds like optimal to use a lookup table in parallel, so such that all of them will produce the same or the result of this lookup table in the same time, because I'm bounded here by the clock cycle. So if all of them can perform the same operation in the same clock cycle, I can generate all bit vectors in the same time, because again, all of them are independent of each other. So I can have many of these lookup table, and how many of them I need? So I need for each bit single lookup table, right? So as the equivalent to the length of the vector multiplied by this value, I need this much of lookup table per problem, which is the pre-aligned filtering problem. So every 2 strings, I need this many of lookup table. Now, if I need more parallelism, I could fit more cores, meaning that each core can process 2 strings, so I can fix another core that solves 2 more strings, then 2 more strings, 2 more strings. As long as my FPGA can accommodate more cores, then I can fit more cores to do parallelism with this processor. And this is what we actually did. This is the gatekeeper, and we have many cores, and I get a read and a reference segment of equivalent length. So I have here 2 segments coming to the gatekeeper core, and another 2 independent, or it could be shared with the same read, for example. So you don't need to transfer the read maybe twice to the core, but you could transfer it once because it's shared between these. So this depends actually on the index you use. Some of the hash table upload 1 read, then query it in the reference and get many location. Then you send only the read and then many of these locations or many of these segments to the FPGA. And some of them do the opposite, upload chunk of reads and then have the hash table entry by entry checked for these segments of the references. So it really depends on the algorithm how you upload this data, but again, you need to investigate that to find the optimal way to transfer the data. doesn't make sense that you always transfer 2 strains per core all the time to the FPGA. If the read is similar or the algorithm generates for you one read at many locations, then you need to transfer that read only once to the FPGA. Another setup could be that you have the reference genome inside the FPGA. And whenever you get the location, you only send the locations in integer format to the FPGA, for example. Then you retrieve the reference segment from the memory of the FPGA internally. In this case, you don't need to always transfer the reference segments to the FPGA. But we didn't investigate all of this because this is not related to Gatekeeper. This is more or less related to the read mapper that you are using or you are trying to accelerate. What we care about for Gatekeeper is to do the operation inside. How we transfer this data to Gatekeeper, this is up to you or up to the user who's using Gatekeeper in the middle of the read map. But it will be really nice to investigate all of these kind of different design choices. However, in the time of the developing Gatekeeper, there was a PCI Express driver. PCI Express is the link used between the FPGA board, which is, yeah, this FPGA board, there is a PCI Express card here, and then from this, you bring a cable to the motherboard, such that you can transfer data from the computer to the FPGA, right? And this is what we call a PCI Express, which is the name of the communication link between the FPGA and the motherboard. And for that, you need a driver. Driver meaning that some APIs where you can use in your program to send some data to the FPGA and receive data from the FPGA. And at the time of developing, around 2016, we used something called RIFA, which developed, I think, by University of California, San Diego. I think these days it's not alive anymore. It's already dead, and by that time they didn't support this board with fully bandwidth of the PCI Express. They only support 4 lanes with PCI Express 3 or PCI Express 2 with 8 lanes, which is really not fully occupying the entire communication bandwidth. And that was a real problem for us, because we are limiting ourselves with the with this bandwidth. We could only transfer this much of data to the FPGA. And this wasn't enough to fully occupy the entire FPGA chip. So we only occupy a small portion of it. Though the utilization of the single core was nothing, almost less than 1% or less than 0.5% of the lookup table available to us. So we could fit 100 cores or 200 cores inside the FPGA, but what we are able to fit only 16 cores. Why is that? Because the data coming from the CPU to the FPGA was limited by the communication channel. So these days, I think we have more drivers that leverage the full bandwidth of the PCI Express. So you could push these boundaries if you have this driver and more advanced FPGA, for example. Anyway, we are not using here any IPs from the company or for the FPGA that limiting you for not using other FPGAs. So our design is pure based on lookup table, that's it, which is the essential part of any FPGA you have. So you can implement the same code without any changes to another FPGA board. And the source code is already available online, so you can have it. And these are the results that we got from the Gatekeeper. So this is the paper, and this is the similar version of Gatekeeper, but implemented on SIMD instruction, called shifted Hamming distance. And actually, Gatekeeper accelerates this paper. So this paper comes first in 2015, then Gatekeeper comes in 2017. So both of them are available online, both of them, the source code in SIMD and FPGA are available. But the SIMD version of it is limited to 128 characters for read length.
Can I ask you a question?
Yes.
Is there any chance that you can utilize more FPGA lookup tables by using the new architecture, like Xeon plus FPGA or power CPU with an FPGA where the communication synchronization costs between the two elements are shortened?
Yes, definitely. Definitely. That's the way to go to improve this work.
Have you ever tested that?
No, not yet. Not yet.
Thank you.
So the best way to optimize this work is to improve the data bandwidth of this communication channel. And that's why Intel, for example, proposed hard architecture where you have the FPGA connected directly to the memory, for example, so you don't need this PCI Express hassle to handle yourself. So you really can get very high bandwidth from the memory to the FPGA board. Or you can think about the latest FPGA board that has HP MEM cube or 3D stacked memory inside the FPGA, where you have a huge amount of data bandwidth to transfer to the FPGA board and do processing. And this is one of the directions need to be discovered to accelerate this work. Because these are highly parallelizable, for example. When you compare it to the dynamic programming algorithm, for example, you cannot parallelize it as much as doing k-alignment filtering. Yeah, but think about, even if you use SIMD instruction, right, to accelerate dynamic programming algorithm, and think in the same method, same headset, like same mindset about gatekeeper. You use the SIMD instruction here, and you use SIMD instruction, which will provide you more speed. Of course, filtering, right? Because you have way more parallelism here at all levels, within the same vector and with all vectors together, right? No, in CPU. I'm talking about CPU. CPU to CPU comparison. You have SIMD instruction and SIMD instructions here, right? If you want to implement both of them and do fair comparison between them, you always find filtering much faster than dynamic programming. Because even at the output level, what you are caring about gatekeeper, it will only give you 1 or 0, accepted or rejected, that's it. But for dynamic programming, you are generating cigar string, you do backtracking and all this stuff, right? Which are not used in filtering.
Applying short extension with assembly parallelized one for filtering. Can it be as fast as shifted having distance implementation?
So, I didn't study both of them, but think about the parallelism you can do. I don't think dynamic programming can be faster with the same implementation, same instruction, instruction set version. If you use SSD4 or SSD1 and so on, right? So it should be filtering always faster.
Or even if the filtering, even if the dynamic programming extension is designed for filtering.
Oh, you mean combining both of them, filtering and dynamic programming?
Yeah, I'm wondering we can combine lightweight extension, which compute somewhat more precise score. If the score reach higher than some threshold, where the score is the same one as used in the final extension, we can distinguish if the C is true or false more precisely than bit vector-based methods. I wonder.
So is this global alignment or local alignment? Because it really matters. So if you use a global alignment,
it should be semi-global one.
Yeah, for semi-global it might work because you don't necessarily need to wait until the very last cell, right? As I presented before. This is the challenge only affecting the global alignment because the result will only be in the very last cell, right? But for local alignment, you don't need to wait until you compute the very last cell of the programming table. So you could terminate somewhere in the middle. for example. So in this case, you are applying filtering. So to do fair comparison, you cannot apply filtering on top of filtering. So if you want to compare gatekeeper with dynamic programming, you need to remove the filter from the dynamic programming, then compare them together. Or apply gatekeeper with dynamic programming and your extension with dynamic programming. Then you do the fair comparison and tell me the results. Okay? It might be same speed up, I don't know. I didn't try. And it might be filtering faster in this scenario because you're already including your own filter there, right? But you need to be careful because in dynamic programming, what you are calculating is integer value, right? With many bits. And this is what limiting dynamic programming from being more accelerated. But here, every entry of the dynamic programming table is single bit. That will provide a huge amount of speed up, right? But there, if you want to use 32 bits for each entry of the dynamic programming table, then think about one row of the dynamic programming table, how many bits you need. And that will affect, for example, the cache line. When you retrieve one line from the cache and reading that at the CPU side, then do some processing. Then one row of the dynamic programming table may not fit one cache line. Then you need 2 cycles to retrieve the result from the cache.
Situation, we can keep entire DB table from designing registers. Can be faster.
Yeah, but still limited by the register size, right? 512, 128.
Here, we don't have to do trace count on the matrix because we only need to do filtering. We can discover the computed TP variables.
But what if it is a good match? Then you need to recompute the things. How do you do backtrack?
We have to recompute the cells.
Yeah.
We don't have to do trace count basically because it is for filtering.
Yeah, this is for gatekeeper, but if you want to do it within dynamic programming algorithm to do filtering inside it within the same algorithm, then you can't just ignore the backtrack because it might be a good match unless you want really to recompute everything from scratch if it's a good match. Then this might work. But you can see about the computation you're repeating. So again, at the end, this is design choice, right? It's up to you to have different parameters, different designs, and then study which one provides you the most speed up. But the best thing about this is that every single character is represented by a single bit. So you can see a lot of computation you can perform within maybe singles in the register, for example. You don't need any of them. So you can leverage the other registers for other comparisons, for other 2 strings to be compared. Again, but this is limited by register size and it has its own drawback as well. So it's not the best always, as you can see here, he's limited to 8, for example. Okay, can we do better? How about scalability? So why we do AND operation at all, the entire read at once, for example. And that opened up for another word called shoji. Yes? A question?
Yeah, I saw the paper 3 days before.
Okay. Okay, so this is shoji paper. We presented this year in Bioinformatics Journal. And what we said, okay, the correct answer always, as you saw, that these green segments, right? The green segments I highlighted in the bat vector masks. And we want to collect them. We want to track these green segments, the consecutive zeros, right? So we said doing AND operation with many other vectors is always inefficient, right? So instead of doing AND with all vectors together, no, go to each vector and count how many consecutive zeros we have in each vector. And this is as simple as this. So we use overlapping slide window, that's why we call it shoji, to quickly and accurately find all long segments of consecutive zeros. And by doing that, we achieve 160x faster than its CPU implementation. And we are able to accelerate best performance CPU read alignment for idlib, which published in Bioinformatics 2017, by up to 18.8x. So this is a dynamic programming algorithm providing global alignment, end-to-end. So we are able to accelerate it by this much only using the FPGA with a new algorithm. So how the algorithm works. So this we all agree how to build it, right? There's no differences here. So either you think about it horizontally or diagonally, same way. This is an exact XOR operation. This is one shift and then XOR. This is two shifts XOR. This is three shifts XOR. And this is the opposite with three diagonals. And all of them independent, so you can produce them right away using lookup table. You don't have to do it cycle by cycle. And now we pick a window size of 4, and then we search each diagonal or each bit vector, and we count the number of zeros. So in each diagonal, we count the number of zeros, and we find that this is the largest diagonal having 4 zeros. So we greedily pick the one with the largest number of zeros. So we said, okay, let's pick this one and record it somewhere. So we record 4 zeros here, and we keep repeating this using overlapping search window. And we keep counting and recording the value we got. So because it's overlapping, so you can see here there are 3 bits overlap between the results of each one. So how we break the ties between these two windows. So again, we pick a greedy choice to store the segment that increased the number of zeros. So you have here 3 zeros and 1, for example, and the previous one was 4 zeros. So which one increased the number of zeros within these 3? So here we don't have any problem, right? Because all of them having 3 zeros. So the first one we have 3 zeros and the second one 3 zeros. So just store it. But if this one has 1 here, for example, and this 3 zeros, then pick the new one and replace the 3 old bits. Why are we always looking for largest number of zeros? This greedy choice guarantees for us to have always 0 false negative. Anyway, whenever you're reducing the number of ones, you guarantee that you are underestimating the number of ones. But we don't want to underestimate it too much. Otherwise, you'll have a huge amount of false positives. And we keep doing this for all search windows until we get this result, which is what we are looking for. And then we count the number of ones. We'll tell you how many differences between the string. Now, how is this different from Gatekeeper? In Gatekeeper, you do an AND operation to the entire vector with all vectors. So that is a way to go if you want to parallelize Gatekeeper more, because here, each search window, it's independent of all search windows. So that will relax the limitation for your read length, for example. So you don't need to accommodate the entire read in one SIMD register so that you can do AND operation together, or many registers, for example. So here, you just need the window to be computed independently. So you can calculate it wherever you like, GPU, FPGA, or SIMD, and there is no limitation to any register here.
I don't understand. An operation can be done bitwise, right? Of course.
Yeah.
For very long.
Yeah, but there's a difference between requiring the entire thing to be exist in the same register when you do AND operation, and there's a difference between only having 4 bits exist in the same place.
No, we can split a long vector into, let's say, 8 shorter vectors.
Yeah, that's a good strategy to do it, but how you are going to implement this, for example, when you amend the zeros into ones, when you flip the zeros into one. If you break it here, or if you break it here, it's really matters, right?
I see.
Because otherwise you need to do overlapping.
I see.
If you do overlapping, yeah, you can fix this problem, but think about overlapping, you're wasting more resources to leverage more SIMD registers, for example. So in here, we are solving this issue by having only 4 zeros need to be exist in the same computation unit, regardless whether it's SIMD register, FPGA, GPU, and so on. So yeah, if these number of differences this kind of threshold, then this is a good match. Keep these 2 strings, send them to dynamic programming algorithm. And this is why we call it the Shuji. And we did some analysis why we pick a window size of 4, not 3, not 4, not 5. And we find 4 is the optimal, and it has an explanation, good explanation behind it, because the minimum number of matches is 0, right? And to be minimal, it should be 1 around it. Otherwise, it's not minimal. And to cover this, we can have a window of size 3, so that we can count these zeros correctly. However, we find a lot of 1, 0, 1 pattern, as we observe in Gatekeeper and here. We can see there are a lot of them everywhere, which are not necessarily part of the solution. For example, this pattern, 1, 0, 1. So to exclude that, we need to increase by 1 bit, such that we include a 0 from the correct ones, for example here, or here. If we include 0 along with this diagonal, then we can ignore this count and we consider this count. That's why we decided to have a window of size 4. Any question? Okay, so we implemented an FPGA, and apparently you are counting the number of zeros, right? So if you want to implement it as a counter in the FPGA, it will be time consuming and not efficient. Why is that? Because a counter in FPGA is implemented as D flip-flops. If you are counting 4 bits, then you need 4 D flip-flops. And that will cost you 4 clock cycles to count a bit vector of 4 bits. However, because we limit it to 4, we really don't need to implement the counter as D flip-flops. What we can do is just a lookup table. So we store these values, and we store the number of the zeros in each vector. And you can't get out of this, right? Whatever you have a bit vector should be one of these values. And the number of zeros is already pre-computed, so you can get it within a lookup table. And that will help you a lot to achieve all the computation in a single clock cycle, rather than waiting 4 clock cycles to compute or just counting. And in our FPGA, we have around 1 million lookup tables, so it's fine to use many lookup tables for each vector, for example. And this work also is available online. You can access this link and download the FPGA source code, which is written in Verilog. And we have, I think, the CPU implementation of it. You can have it, you can try to change it, increase the window size, do some modification and figure it out, and maybe propose your own idea to improve this work. But we didn't implement it in CPU. So this is another future plan if you would like to improve it.
What do you think of adopting Chisel or Scala-based HDL?
I didn't use it before, honestly, but if it is higher level synthesis tool where you can write in C, so it might not provide you same performance as Verilog, because Verilog is the lowest level. Same as you write in assembly or you write in C or in Java, right? So I always prefer to go with system Verilog or Verilog. But you could go with higher level synthesis tool.
Chisel is not like kind of implementing hardware with C or Java or something else. It is a kind of wrapper, or the Chisel engine transpires somewhat functional code into very backend.
I see.
I heard RISC-V processor is basically designed with Chisel, and it was very successful supporting multiple configurations, such as 4-bits or Q-links or something else. I wonder, changing the bit lengths or some other parameters in this algorithm is somewhat hardiness when implemented in Verilog. And yeah, what is the reason I wanted to ask you?
I see. So basically, maybe I used Verilog because I'm very good at it, so I feel comfortable programming using Verilog or system Verilog. But yeah, why not using another language? It doesn't matter as long as you achieve the same performance, for example, as Verilog. Or I'm not really sure. I didn't use Chisel before. I don't know about it, unfortunately. But this code is really simple in Verilog when you implement it. When you read it, you will see it as exactly as you implement it in C, for example. There's no real hardware behind it because the algorithm itself is very simple. You're just looking for 4 bits and that's it. So you design these blocks that count 4 bits and you duplicate them. You have a lot of replications everywhere. And then you distribute it over the read lane. That's it. So you could read the C implementation as well of this algorithm. You can understand it better and implement it the language you like. But for example, if you would like to go to customized CMOS chip, so if you want to implement the same design but for CMOS chip. So I'm not sure if Chisel would support that. For Verilog, it's highly supported by Cadence tool, for example, if you want to implement it as a chip. So you just put the Verilog code and done. Then you compile, synthesize, do all the process as usual, and you will get the RTL for the chip design or the layout of your chip.
It's almost comparable to our generate CMOS design. Or
So I think
Maybe?
I think the second author, Hassan, should be very familiar with Chisel, so you should send him an email.
Okay. Okay. Alright, so the third work as a pre-alignment filtering. We call it Sneaky Snake. And this work we implemented for CPU, GPU, and FPGA, all available platforms. So this work is available in archive, but we didn't publish it yet. We submitted it and it's under review. So again, we observed the same thing, that correct alignment is a sequence of non-overlapping long matches, so long consecutive zeros. And still the same goal, we have to track these consecutive zeros. So you could have different method, different algorithm, more accurate, more faster than whatever we presented here. So think about it a way of doing that and implement it yourself and check whether it's working or not. So the key idea, we said, okay, approximated distance calculation is really similar to single net routing problem in VLSI chip. What is single net routing problem is as simple as this. Think about having a chip. So a chip is anything in electronics. Could be in your phone, could be in laptops, anything you see this black piece. The black piece, you see legs, right? Which are the input and the output of the chip. So sometimes we need to add some more component inside the chip or outside. So think about you have another chip here and you want to communicate with that chip to the other side. So you have an input here and the output here. And you want to add a wire or whatever routing resource you have to connect these two things. However, when you go from right to left, you have a lot of obstacles. You have a lot of components inside the chip. As a module, or as a processing unit, inside the GPU, inside the FPGA, whatever you have as a chip, it should have many modules, right? And your goal is to avoid these obstacles such that you have a nice routing path, for example. Could be shortest path, could be a path with less obstacle, or it could be a path with less latency, for example. If you can pass through module, it will be slower than passing without a module, for example. So you can think about it a different way. And this is the definition of single net routing problem. So these are the results, and if you check when we implement it in CPU, there's no SIMD acceleration, nothing, just pure C code. We got 37x speed up compared to IDLO. And with the hardware acceleration, we got this much of acceleration using both GPU and FPGA. We find GPU performance is really close to the performance of the FPGA. And there are little fundamental difference between them. And the difference is that, as I told you about the configurability. or parameterizable your design. So if you have a distance threshold of 10, now you want to change it to 15. Or if you want to sweep the edit distance from 0 all the way to 100. In FPGA, it's really challenging because at each edit distance threshold, you need to do some changes to your redesign, then synthesize it again and implement it in FPGA. But for GPU, you can do it as a for loop and sweep as much as you want. And you have the same design, is not changed. So, how we do that? Again, the same neighborhood map, everything here same, but think about now we want to lay out horizontally, just to make a simpler visualization. And now we want to change the 1 into a black box, and the 0 into white box. Just changing the same property, just a way of interpreting it. We're not changing the computation. So the black will be an obstacle, and the white will be an available path for your routing resources. And now we have the entrance at this side and the exit at this side. Doesn't matter where you have it. It could be the other way around. Should be correct. And now we set our threshold 3. That's why we have 7 rows in this matrix. And now this is your snake, and want to go through this maze all the way to the exit. So what we have to do is check row by row. How many steps we can go further in each row. So the first one, 0, 0, 0, 4 steps, 1 step, 0, and single step. So as a wise man, you should pick this path, right? Because to provide you with furthest as possible to travel, right? So we pick this as our initial path. So we pick 4 steps plus 1 obstacle such that we can avoid it and move further. And then we reduce the available obstacles by 1. So we have now 2 obstacles that we can avoid instead of 3. And now we repeat this exact same steps. So we check how many steps we can move further in each row. Now we have 0, 0, 4 steps, 0, 2 steps, 0, 0, 0. And now which one to pick? Of course, the largest number of steps. So we go 4 steps plus 1 obstacle, and we reduce the number of available obstacles. Now we have single step or single obstacle that we can avoid. And we repeat exactly the same step, but you can see there are ties. There are 3 available paths. Which one to pick? Doesn't matter. All of them will achieve the same result or reach to the same destination. So we pick any one of them and we reduce the number of obstacles to 0. If we reach the obstacle to 0 and we didn't reach the exit, probably there are too many obstacles in your path, and these 2 strings have high dissimilarity, so ignore them. If we reach the exit and the obstacle is 0 or more, then this is a good match, then keep it and send it to the dynamic programming element. However, if you think about the computation, it's more or less same as Shuji, for example. But the fundamental difference here is that you are not breaking the exact match. You are keeping as it is. And that will give you a lot of reduction in the false positive. Really, you significantly reduce the false positive. Because if you break these in 2 windows, then you might pick those as exact matches and those as exact matches. And think about also if you break it at here, you might take as this exact match and then this as exact match, and then you might ignore one of these mismatches. So you are underestimating the number of matches. However, this is not the only difference between the other filters. The big difference is this. This is what you really need to compute, not that. Why is that? Because when you compute it, you go to this row, you find it an obstacle, so you don't continue computing. You just terminate the computation here. You move next, you terminate the computation here. Here, it's available path, so you keep computing until you find an obstacle and you terminate. So think about the computation saved here, 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 and there. And think about it at the scale. When you save a lot of computation by not computing all of these bits. So what you compute is very few bits of the entire matrix. Now compare this to dynamic programming algorithm, where you have 32 bits in each cell. You have a lot of reduction in terms of computation compared to dynamic programming algorithm. And this is the beauty of having realignment filtering. You can play whatever you want with it. You can assume everything as long as you don't have false negative. So always underestimate the number of differences and do whatever heuristic you need to do, which can be beneficial for the hardware you have or even for conventional CPUs. Now the computation here are also done on the fly. You don't need even to store this. What does that mean? You can go to the first character of the first string and the first character of the second string and compute this. Then the first character of the first string and the second character of the second string, and you compute this cell. You can repeat doing this just by checking if this is equal to this, move on, if not, stop. So you don't need even to store it. And that's what caused the CPU implementation of this, without any SIMD, without any vectorization, to have this a huge amount of speed up, 37x compared to dynamic programming, single-threaded, single-core, everything fair comparison between the two tools.
Did you compare this algorithm to a class of algorithms which is denoted as greedy extension? One example is P-aligner by G. Myers. It adopts an algorithm which is very similar to this greedy extension, the final extension. And the code itself in the P-aligner is very messy and our performance not good enough on modern processors.
Okay.
But I wonder, it could be very informative to compare this algorithm.
Yeah, that's good. I will consider it. I didn't compare it before, but I will consider it. Anyway, whatever, any work, any aligner that use seed extension or chain of seeds, meaning that you have the seed, which exact match, another seed exact match, then perform some local alignment between the two seeds to do extension, for example. This is still more expensive than doing this. Why is that? Because if you notice, when you have these ties, we didn't perform any alignment here, right? So it doesn't matter if you pick this path or that path, right? All of them for us same. And that is way cheaper, computationally cheaper, than doing Smith-Waterman between every two seeds, right? So in theory, it should be faster. And if you check these computations, it's nothing. It's real nothing. It's very cheap to perform. But yeah, I'll consider that. I'll compare it with this work. By the way, for SSD shifted Hamming distance, the paper in 2015, they compare with the G. Myers, and they achieve 7x speed compared to that, which is SIMD version of the gatekeeper. And they compare with that, and we are much faster than SSD and gatekeeper, Shuji, and all those works. So in theory, yeah, it should be faster than the G. Myers. Okay, any questions?
I got a question about checkpoints here. I didn't get the meaning of checkpoints here. So
Yeah, checkpoints
is
it's same as the number of steps you're repeating here. Do you remember this when we have arrows here and then arrows? So how many times you reach an obstacle and then you repeat the computation. So every time you reach an obstacle after the path, you are done with all the computation of this. So you start another iteration or another checkpoint.
So do you mean that when you start checkpoint 2, the best alignment so far is taken and all the others are discarded, or if other traces may reach the checkpoint by using only up to 3 obstacles, then you keep them all?
Mm-hmm. So the only thing we keep after each checkpoint is this counter. Only this counter. So after this checkpoint, we have a single obstacle we reach here, right? So I just need to know how many obstacles I faced to reach to checkpoint same here, same there, until I reach exit. So if I reach exit and I have a negative, let's say, negative counter here, then that means I had a lot of obstacles on my way to reach here. So this is a bad alignment. But in fact, I don't wait until I reach exit to know that. Whenever I reach a checkpoint and I'm having less or more obstacle than the threshold, then I just stop computing and ignore these two strings.
So conversely, if a search bar doesn't have the specified number of obstacles, then you keep it.
Yes,
So you never miss the true alignments.
yes, exactly.
Okay, that's what I wanted to
Yeah.
cover.
So always we guarantee in all our these pre-alignment filtering 0 false negative. We never ignore any correct alignment.
Okay, so probably this algorithm is much closer to the D-alignments wavefront algorithm without any pruning.
Okay, okay. But do they apply any dynamic programming between two seeds or maximal exact seeds? Or?
No. Greedily takes the exact matches until they hit mismatches, in this case obstacles, and then diverts by 1,
Okay.
then goes through until the next obstacles, and the checkpoints are called as wavefronts in there.
Wavefront.
Wavefronts.
Okay, I see. Yeah, I'll check that algorithm. I never studied it before.
But they have lots of pruning algorithms, so this is, this algorithm can ensure the no false negatives.
I see.
So that's a huge difference between their algorithm and this algorithm.
Okay. Yeah, we try always to make it as simple as possible because anyway we don't really need some accuracy coming from here or cigar strain or all those kind of things, alignment score and so on, because we're still using dynamic programming algorithm. You're not replacing it. So the simpler you have computation here, the better for you. And we want to implement it in FPGA, GPU, and all those stuff. So checkpointing is very beneficial for us. Why is that? You can send all this area to a single thread in GPU and compute this area independently from this, independently from that. But some of you ask, how do you know the checkpointing in advance before so that you can split it over threads? We don't know actually. We could have a heuristic, just every 32 bits send it to a different thread, for example. That will affect the accuracy for sure, but will affect the false positive. So here you can have 64 wide window or matrix, submatrix, and send it to thread or FPGA and do it in parallel with other submatrices.
Okay, sorry, then D-aligners algorithm is pretty much different from this.
Okay.
And actually, their algorithm uses the number of mismatches or the edit distance as the checkpoint here. So
Oh, I see.
this is pretty much.
Okay. Okay. Now this is pretty much
related
independent.
to dragons.
Sorry?
Is it somewhat related to dragons? FPGA-based extension algorithm? Not documented as paper, but we can release.
I don't know. Do you know what's the algorithm?
Yeah, detail is hidden, but we can read the submission of patent, US patent online.
I didn't check it honestly before. But if you know the algorithm, maybe you can tell us if there are similarity or differences.
Yeah, I can't remember the most of the details, but the idea breaking the input sequence into chunks and do a kind of grid alignment parallel is
I see.
somewhat similar.
No, for us, we are not breaking it. This is just, I'm telling you a way if you would like to do it. So I'm breaking this in GPU, for example. But in CPU, no, I'm keeping it as it is. Because in CPU, the best for me is not to break it, to maintain the accurate one. Because when you break it, you always expect more false positive when you break this matrix. But for FPGA GPU, yeah, I'm doing that. Because in FPGA, for example, this is more challenging than Shoji. Usually I'm fixing the width to be 4, always. So it's easy for me to design a lookup table that accommodate 4 bits. But here, I'm searching unlimited, right? So my search space is the entire thing, because I don't know this in advance, how many steps I could move. So my search space is from here all the way to there. So I have to design a hardware for that. So either you want to have the entire thing in one register, which is, again, a drawback of Gatekeeper, for example. So how to do that is checkpointing. So you fix the width of this to 32, for example, then it's easy for you to design hardware for 32-bit counter, for example, right? So this is the way to address these challenges in hardware. And when we implement the snake, the version of an FPGA we call it snake on chip, and the version for GPU we call it snake on GPU, and on CPU sneaky snake. So snake on chip has this much of footprint for lookup table utilization. So you can see this single core. You can see all of them more or less around 1% utilization of the FPGA, which is really small footprint. So you can fit hundreds of these cores in the same chip. Or you can use the ultra-scale, more advanced version of Vertex 7, the FPGA we use, and which achieve maybe double number of lookup tables. So you can get 200 cores on the FPGA that you can process. Again, we are able to have only 16 cores because of the PCI Express bandwidth. But this is not an issue in real life. You can address it using many other drivers, or use the driver provided by Linux, or use UltraScale, or use HARP, or use GPUs, for example. So you can always alleviate this problem, or you can get more bandwidth. And this is the link to have 3 design, the CPU, the GPU, and FPGA. Please have a look there and check the source code. We provide the data, the examples, how to run the code. Everything is documented. We normally have a well documentation for all of our software. And feel free to contact me if you have any issue, any bug, any problem, any comments, or maybe collaborating on improving this work. Okay, so what was the problem in all these works? Again, data movement. So moving data from CPU to FPGA to GPU, always a problem. It might already provide you some great speed up by using FPGA and GPU, but we are always hungry for more. If you can achieve more, it will be great. So that will be the motivation for the next work. So we need to design mapping and filtering algorithm that fit processing in memory. What does it mean processing in memory? So if you still remember UPMem, the French startup that I presented their DRAM chip, that you can just plug it in your motherboard and you do computation inside the memory. So this is one example, one of the great example, practical example of processing in memory. And there are a lot of other examples of this technology providing you processing inside the memory. And this is our another work called Gram Filter, where we do filtering inside 3D stacked memory. So maybe it's good for me to jump into 3D stacked memory before explaining the algorithm for those who are not aware of 3D stacked memory. So 3D stacked memory meaning the memory chip itself, the DRAM that you have in your computer, is 3D stacked or integrated to many of them together. So this is a DRAM chip, this is another one, and the very last layer is a logic layer. Think about it as FPGA or GPU or even CPU core. We call it logic layer. You can implement anything here. You can add ARM core, for example. You can have many assumptions for this layer. What is it actually? But those are memory, and the good thing that all of this is on the same die, meaning that you don't need to transfer any data to somewhere outside this chip. All of this in the same component. And those very high bandwidth links, transferring a huge amount of data, more than 10 or 100 gigabytes per second between these layers. So what you can have, you can do parallel processing because we have banks. So each chip here divided into banks. for each bank, you are reading data vertically, bit by bit or row by row, and then you can have some processing here. So you can distribute your data over these banks, over these layers, and the computation will be in the very last stage. So if you can have hash table, for example, divided into this, here, 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 and then you do computation only here, you can achieve very fast computation and very high parallelism without moving any data. And this is what we call 3D stacked memory. So this proposal of 3D stacked memory been there for more than 30 years. It's already something from the past, but none of the companies could achieve it as a product. So it's only achieved recently by this French startup as 3D stacked DRAM. There are some proposals where they have, for example, micron FPGA, where they have HBM cube next to the FPGA chip. But this is a separate board. You can't use it as DRAM in your computer, for example.
How many slides do you have left? 50?
Yes, around.
Okay. Can you be a little bit quick?
Okay, yes. So this is probably the last work will be presented. So this gram filter, we observed that FPGA and GPU accelerators are heavily bottlenecked by data movement, right? So let's try 3D stacked memory without moving data. And by using this algorithm, we achieve this much of speed up, 1.8x to 3.7x, faster than fast hash filter. Some of you could ask me why we leverage very latest technology and we achieve just only 3x of speed up. Okay, I'm going to tell you the reason, but in the next few slides. So what we did is we built our own hash table inside this memory. Why is that? Because we don't want to move the data, actually. We don't want to build the hash table in CPU and then move it back to the 3D stacked memory. So how we build it? We said, okay, let's divide the reference genome into bins, and then represent each bin as similar optimized version of the hash table. How we do it? We pick a length of 5 and we said enumerate all possible value of seeds of size 5. So it could be either all of it A's, it could be all of it 4 A's and the last one C or G or T, all the way to all of it T's. So just enumerate all of them. And then whenever you have A, for example, you record 1. And then the next one you have CCCC, so you record CC1 to them. And you keep doing this until you get this bit factor. And this is what you actually store. You don't store these values. You keep doing this for all bands, and what you got this. So you store B1, B2, all the way until you finish the entire reference genome. And this is some theoretical calculation on the size of bits you need to accommodate in your 3D stacked memory, how large of these. So this is how many possible value you have in each character, 4, right? Either A or C or G or T. And what's the length of this is 5. So how many entries you have this? So because you are smart people, you can calculate it as 4 times 4 times 4 times 4, or 4 to the power 5. And you get the number of bits you can calculate to accommodate this. Times T, which is the size of the reference or number of bits you have for the reference genome. And this is the memory footprint you needed to inside the 3D stacked memory. And then how we do read mapping here. So you pick the read and you get all the seeds of size 5. 5 here is something configurable, so don't think about it as fixed size. It can be 6, 7. And then we already having these, or we generate this from the read based on the matches. We record 1, whatever it says, and we compare it with the bin we stored inside the 3D stacked memory. So this is the index before, and this is from the read. And when we generate bin from the read and bin from the reference genome, we compare them together and see how many of those ones appearing in that bin coming from the reference genome. And then we add them together, the number of ones. And if they are exceeding the threshold, terminate. If they are less than the threshold, keep, and something like that. Okay? So any question for the method? Sounds very easy, right? But what's the bottleneck here? Why we achieve 3x speed up? So the problem is that we don't load all those bends every time we have one read. So when we have read, we don't compare the bin of the read with all bends we have for the reference genome. What we do is we got from the CPU the location that this read mapped to. So we still have another copy of hash table in the CPU that compute for you the location. And then the location will be transferred to the 3D stack memory to tell you what bin to extract from the reference genome and compare it to the bin of the read. And this is really time consuming because we are still waiting for the CPU to process the hash table and transfer this location to the 3D stack memory, though we are handling all of them in parallel fashion. And that's what caused the unexpected speed up we got from the 3D stack memory. Okay, so this is a summary, this is 3D stack memory, and this is what we need as a logic in the logic layer, just a piece of comparator that can compare two bins, then the accumulator that can add all ones, and then just that's it for each logic layer. Yes, this is the folder. You have the source code, you have everything there. Feel free to play with it. And this is a Gen cache. This is a paper presented recently in Micro 52. This is not part of our group, it's not published by ours. This is another work that leveraged our own pre-alignment filters. So they said, okay, you have too many filters, but there's no one best, right? Some of them are very low overhead, but the false positive is really high for the distance more than 8. Then okay, let's use gatekeeper for the distance less than 8. And let's use this if the distance is this. And let's use another pre-alignment filter if read length is this much. So they have too many pre-alignment filter working in parallel. All of them together exist inside the cache. Not memory, not CPU, not FPGA, not GPU, but they choose the cache. For those of you who's wondering what is cache, you can think about it as a memory, but it's way faster than the conventional memory, but the size is really small. And those guys enabling processing inside the cache by adding some computation, some logic layer, let's think about it, inside the cache. So this is not practical because we don't have any cache that enables you to do some computation inside it. So this is more or less some simulation or theoretical paper. But they have a problem that they increased the size of the cache by 16%. Industry people never like this, never like increasing or having an overhead to existing technology, even DRAM, even the cache itself. So this is the little bit the drawback of this work. And this is how many pre-alignment filtering they use for different phases, different conditions. If there are 0, 0, they use Hamming distance. If it's 1, shifted Hamming distance, and so on. And by doing processing in cache, so they don't need to wait until go all the way to processing cores. So from DRAM direct to the cache. So they save midway, for example, energy or the processing time. And they could get this much of speed up compared to GenX. GenX is another proposal for processing the dynamic or the fold read alignment. So this is by having single pre-aligned filtering in cache, and this is by having phasing or many of these pre-aligned filtering working inside cache again. So they get this much of speed up or throughput. Okay, now I'm done with seed filtering and pre-aligned filtering, and the last work, which is also not part of our work, which is called Darwin. Darwin is a recent paper published in S+ 2018. And in Darwin, they accelerate the dynamic programming algorithm, though I told you that we can't accelerate it because you still need to respect row by row, or column by column, or anti-diagonal. And interestingly, what they did, this is again not part of our group. What they did, let's jump into the proposal. Yeah, here they did some theoretical analysis. If we do the dynamic programming algorithm in CPU, we will need this much of resources, this much of cycles, this much of energy. But if we do it in hardware accelerated, we need this much of resources, single cycle, because as I told you in FPGA, we are only bounded by the number of cycles to do computation. But we can increase the lookup table utilization. So you can increase 1,000 of them or 1,000,000 of them, doesn't matter, as long as you have this much of lookup table. All of them will be computed by single clock cycle. And they require this much of energy. You can see the reduction is really huge in energy and the number of cycles. What they did is they said, okay, let's divide the dynamic programming tables into tiles or sub-matrices and then process each one individually using the chain of lookup table, or what we call a systolic array. So I'm not sure, this is questionable about the accuracy of this algorithm because they are dividing the dynamic programming table into sub-matrices. So I'm not sure, for example, this row, the row here, how they consider the rows there, the result there, because this cell to be computed, we need to have this cell and this cell and this cell. So for example, how we solve this issue, I'm not sure. Either I didn't understand it from the paper or they didn't present it in the paper. And they implement it as a CMOS chip. So it's not FPGA. They have some FPGA results, but the final product is CMOS chip. So it's more specialized than FPGA, for example. And they get this much of speed up compared to graph-based alignment, and this much compared to idlib, which is around 5,200x speed up. Of course, this is a simulation result because they did CMOS, they didn't fabricate the chip. But you can think about compromising the accuracy while gaining some speed up. It can be beneficial, for example, metagenomics application where you don't need really accurate CTAR strain for classifying the genome you have in hand. And this is, as I told you, it's not part of Safari group, but we have bitmap work, which is under review now, and we did the full dynamic programming inside the memory rather than filtering. We are developing our own algorithm based on BiTab, which is 30 years old. This BiTab algorithm was published in the old days, but has no backtracking step. So what we did, we proposed our own backtracking step for that algorithm, and it wasn't used as Smith, Waterman, Needleman mentioned, all those algorithms, because it requires a lot of memory to do the backtracking and all those things. But since we have a huge amount of bandwidth inside the 3D stack memory, we could be able to implement it inside 3D stack memory. And we achieve a better performance than Darwin did. And this is a conclusion in all these three directions to accelerate read alignment. So read alignment can be substantially accelerated using computationally inexpensive and accurate pre-alignment filtering algorithms designed for specialized hardware. And I need to really emphasize that. We really need both of them. Algorithms and hardware. Please don't try to accelerate using only hardware. Try to tweak or study the hardware you have in hand and then do some changes to the algorithm such that you leverage the full power of the hardware you have in hand. All the three directions are used by mappers today, but filtering has replaced alignment as the bottleneck because it's the first self-defense, let's call it, where you reduce the entire workload and send only a few of them to the dynamic programming, which is the most expensive part. So pre-alignment filtering doesn't sacrifice any of the alignment capabilities. By not accelerating the dynamic programming part in FPGA, for example, you are not limiting yourself by certain alignment score or certain scoring, because you are not limiting the number of bits for each cell. For example, 32, 16 bits, 5 bits. You don't have this problem when you implement it in CPU, for example, not SIMD, of course, not other choices. So by doing pre-alignment filtering, you can do whatever you want with it. There are no restrictions, and you're not sacrificing any of the alignment capabilities, as it doesn't modify or replace the alignment step. So please keep pushing the pre-alignment filtering to get the best performance out of it compared to the dynamic programming algorithm, such that your tool will be used for many biologists, many bioinformaticians who need a wide range of alignment score, for example. Okay, where's read mapping going next? This is the last step, and I'm having just 2 or 3 slides here. Now think about sequencing machine. Actually, the entire read mapping problem came because of the sequencing machine are not able to read the entire content of our DNA, right? Now think about the future, 20, 30, 50 years later on. If these sequencing machines are developed such that they produce 100% accurate. There's no sequencing error. And each read is genome long read. So think about this. This is really very ambitious. What will happen to the read mapping? What will happen to all the work we propose, pre-alignment filter, FPGA stuff, and all those things? Think about it. Do we still really need read mapping? You need to think twice before asking this question. Think about metagenomics, plant genomics, all those research fields where we compare genome to genome, not read to genome, even genome to genome. When you compare genome to genome, you don't expect to have few snaps or a few mismatches such that you can use hand distance, for example. You still need the dynamic programming part to compare this genome to that genome such that you can find insertion, deletion, could be 500,000 insertion or deletion, right? So you still need the dynamic programming algorithm. And to do that efficiently, you still need the pre-alignment filtering to tell you whether do you need to perform dynamic programming algorithm or not, right? So in this area or in this field, read mapping won't die. It will still alive there, wherever we compare genome to genomes. So the future of this area or hardware accelerated should be there, but yeah, there's papers showing that Africa have more things than the reference genome we have. That's why we need for each population to represent them using a unique reference that represents that population. And for that, we'll keep comparing the genomes together, African with the European. Even within the same country, we keep comparing the genomes to the other species. So did we achieve our goal? Did we get this smartwatch by doing all those works? Of course, no. That's why we need a specialization. As Tesla, as Surpass, as Preferred Network, as all of these companies providing this specialized hardware to do genome analysis. Unfortunately, we don't have any companies that start this initiative, for example. They always start doing dynamic programming on FPGA, but not the entire read mapper. Did we answer our question? How, where to enable fast, accurate genome analysis with privacy preserving, exascale, all of these? We can't answer this until now. And we don't know the answer where to place this, but what I know from my personal point of view is we really need to start to do some processing inside the sequencing machine rather than our CPUs. And by the way, most of these Illumina machines have their own FPGA card inside the sequencing machine. And as you can see from our pre-alignment filtering, they have very minimal lookup table utilization, less than 1%. So you can fit our design inside this machine if their code is transparent to us such that we can modify it and upload it to this machine. So this is the conclusion. System design for bioinformatics is a critical problem. It has large scientific impact on the society as well. And we show that accelerating key step in bioinformatics, such as read mapping step, there are too many other steps, such as assembly, genome polishing, and all these need to be accelerated as well. But this is the first step that is common between all the analysis pipeline and bioinformatics. There are many bottlenecks, and we try our best to accelerate it using pre-alignment filtering. And we cover recent ideas that we just published this year, previous year, and years ago to give you just some insights, some hints about the future of this. And we started this, Professor Mutlu, Professor Jalal Khan, they started this around September 2006. Me, myself, I started this area in 2014. And I would like to acknowledge Professor Mutlu, Jalal Khan from Bilkent University, many of my friends and colleagues, and funding agencies, and all source code we have available in this link. Feel free to access it, as well as reporting bugs, use the code, improve it, and suggest some improvement on these projects. These are two books where you can read more information about all these algorithms, where you can get some background. I really highly recommend these two books for you. All right, thank you so much, and any questions?
The last one, FPGA-based program implementation.
No.
Is the paper available?
Yeah, Yeah, the paper is available. They have two versions. They have Darwin and Darwin Whole Genome Sequencing, WGS. Two versions. Two papers.
I'm very curious about your implementation.
Which one?
Yours.
Bitmap.
Yes.
Oh, Bitmap is still under review. We just submitted last month. So it's not available anyway. It's not available anyway. But we will be happy to talk about it if you need more information how we implement it. Yeah, we can share some slides with you. Yeah.
How much will it cost for buying 3D structured memory with logic in the next, say, 5 years, 5 to 10 years?
So, yeah, good question. I have no idea about it.
Obviously, it's so expensive, so
Yes, it should be.
the biologist will not want to use it right now, but in the near future, the cost will be reduced to some extent. So
Yes.
what is your idea of the cost?
Yeah, it's okay. We can afford Tesla car with $100,000, while a normal car is $20,000, right? So it's always the nice thing coming at cost, right? So why not to try with the genome analysis? I think genome analysis is as important or more important than autonomous car, right? So why not paying more extra cost, but doing the analysis at scale? Think about hospitals where they have critically sick children, where they really need to sequence their genomes as soon as possible and give them some personalized medicine. So paying extra money is not a problem always when we come to specialization. And for this 3D stacked memory, you buy them once and then you reprogram this logic layer, for example, if it is FPGA. So you can use it for other applications. It's not necessarily just to be dedicated for genome analysis. So you can reconfigure this hardware to be set in a cluster, for example. Maybe Amazon will have such cluster.